What is the concept of spontaneous generation? Living organisms could develop from non-living or decomposing matter. We challenge the concept of spontaneous generation by showing that maggots on decaying meat came from flies, not from the meat itself. Francesco Redi, who showed that mutton broth boiled in flasks and then sealed could still develop microorganisms, which supported the theory of spontaneous generation. John Needham, who showed that flasks sealed and then boiled had no growth of microorganisms. Lazzaro Spallanzani, who proposed that air carried germs. Spallanzani, who disproved the theory of spontaneous generation. Louis Pasteur, who demonstrated that dust carried microbes and proved evidence for the existence of heat-resistant forms of bacteria. John Tyndall, who showed that a silkworm disease was caused by a fungus. Agostino Bassi, who demonstrated that the Great Potato Blight was caused by water mold. M.J. Berkeley, who showed that the pebrine disease of silkworms was caused by a protozoan parasite. Louis Pasteur, who developed a system of surgery designed to prevent microorganisms from entering wounds. John Lister, who transformed the practice of surgery. John Lister, who established the relationship between Bacillus anthracis and anthrax. Robert Koch. What are the Koch postulates? Limitations of Koch's postulates. Some organisms cannot be grown in pure culture using humans. In completing the postulates is unethical. Molecular and genetic evidence may replace. What vaccination did Edward Jenner make? Smallpox vaccination. What vaccinations did Pasteur produce? Chicken cholera, rabies and anthrax. Who induced formation of diptoxin antitoxins? Rabbits. Emil von Behring and Shibasaburokaitis Otto. Who showed that the silkworm disease was caused by fungus? Bassi. Who demonstrated that the Great Potato Blight of Ireland was caused by water mold? M.J. Berkeley. Who showed that the pebrine disease of silkworms was caused by a protozoan parasite? Louis Pasteur. A simple morphology and lack a true membrane delimited nucleus. Prokaryotes. What is a morphologically complex and has a membrane enclosed nucleus? Eukaryotes. What led to the discovery of viruses as disease-causing agents? Bacterial filter that removed bacteria and larger microbes. The five-kingdom classification scheme is the Monera or Prokaryote, Protista, Fungi, Animalia and Plantae. What are ribozymes? RNA molecules that form peptide bonds and perform cellular work in replication. The three-domain scheme is bacteria, archaea and eukarya. The three-domain scheme is based on a comparison of ribosomal RNA. The earliest cells may have been RNA surrounded by liposomes. What are the three bacterial morphology? Bacillus coccus spirillum. What are the types of light microscopes? Bright field microscope, dark field microscope, phase contrast microscope, fluorescence microscopes. What is a measure of how greatly a substance slows the velocity of light? Refractive index. The direction and magnitude of bending light in a microscope is determined by the blank. Refractive indexes of the two media forming the interface. Which microscope produces a dark image against a brighter background? The bright field microscope. What is the product of the magnifications of the ocular lens and the objective lens? The total magnification. What is the total magnification of the bright field, dark field and phase contrast microscopes? 2000x. What is the total resolution of the bright field, dark field and phase contrast microscopes? 0.2 What is the purpose of the bright field microscope? For live and preserve stained specimens. What is the purpose of the dark field microscope? To observe living, unstained preparations. Which microscopes are good for observing living cells? The differential interference contrast microscope, phase contrast microscope. Which microscope enhances the contrast between intracellular structures having slight differences in the refractive index? Phase contrast microscope. Which microscope shows a bright image of the object resulting from the fluorescent light emitted by the specimen? The fluorescence microscope. The fluorescence microscope exposes the specimen to what types of light? Of violet or blue. The distance between the center of the lens and focal point? Focal length. The shorter the focal point, the blank the magnification. Greater. The shorter the wavelength, the blank the resolution. Greater. The distance between the front surface of lens and surface of cover glass on specimen. Working distance. As the working distance decrease, the field of view blank. Decrease. As the field of view decrease, the light gets blank. Dimmer. What is the purpose of adding the immersion oil? To obtain more light by providing the same refraction energy as glass. As the magnification increases, what happens to the focal length? It decreases. What is the charge of a bacteria cell surface? Negative charge. What is the charge of basic dyes? Positive charge. What is the charge of acid dyes? Negative charge. A thin, uniform film of bacterial growth on a glass slide in order to proceed with further staining for microscopic examination. Smear. The process by which organism is killed and firmly attached to microscope slide. Fixation. Heat fixation versus chemical fixing. Heat fixation preserves overall shape but not internal structures. Chemical fixing protects fine cellular structure and shape of larger, more delicate organisms. What information does differential staining provide? Divides microorganisms into groups based on their staining properties. What is the most widely used differential staining procedure? Gram staining. Negative staining. The bacteria is not stained, the background is stained. What is shadowing in electron microscopy? Coating specimen with a thin film of heavy metal. What is the total magnification of the transmission electron microscope and the scanning electron microscope? 1,000,000x. What is the total magnification of the scanning electron microscope? 1,000,000x. Which microscope uses electrons reflected from the surface of a specimen to create an image? Scanning electron microscope. Which microscope produces a three-dimensional image of specimen surface features? Scanning electron microscope. In which microscope do the electrons scatter when they pass through thin sections of a specimen? Transmission electron microscope. Which microscope uses transmitted electrons to produce an image? Transmission electron microscope. Spherical or ball-shaped? Cocci. Rod-shaped or cylindrical? bacilli, corkscrew-shaped, spiral. When two cocci bacteria are stuck together, it is called blank. Diplococci, a chain of cocci. Streptococcus, cocci in a grape-like cluster. Staphylococcus, Vibrios are curved rods. Describe the spiral morphology. Rigid bodies move by flagella. Spirochetes morphology. Flexible bodies move by axial filaments. Organisms that are variable in shape are called pleomorphic. About 70% of antibiotics come from what genus? Streptomyces. What is the purpose of the prokaryotic cell membrane? It separates the cell from its environment. It serves as a selectively permeable barrier. Archaea membranes have what type of phospholipid layer? Monolayer. What do most bacteria cells lack? Sterols. What is the most widely used model of membrane structure? Fluid mosaic model. What is the phospholipid of Archaea? Alglycerol. What is the phospholipid of bacteria? Diglycerol. What is the purpose of the complex foldings of the plasma membrane in photosynthetic bacteria? To increase the surface area available for photosynthesis. What type of plasma membrane do bacteria with high respiratory activity may have? A membrane with intensive foldings. Macroalmonds. COH, NSPK, Ca, Mg and Fe required in large amounts. Microalmonds. Mn, Zn, company, Mo, Ni, and Q required in trace amounts. Requirements for carbon, hydrogen, and oxygen. Often satisfied together, autotrophs UC as their sole carbon source, heterotrophs UCO2 as their C source. What elements are needed for synthesis of important molecules such as amino acids, nucleic acids? Nitrogen, phosphorus, and sulfur. What is the use for amino acids? For protein synthesis. What are purines and pyrimidines used for? Nucleic acid synthesis. What is the function of vitamins? They function as enzyme cofactors. Bacteria can only take in blank particles across a selectively permeable membrane. Dissolved. All microorganisms use what type of transport mechanisms? Facilitated diffusion and active transport. What type of transport do bacteria and archaea use? Group translocation. Which microorganism only use endocytosis? Eukarya. Molecules moving from regions of higher concentration to one of lower concentration between the cell's interior and the exterior is called blank. Passive diffusion. Which molecules can move by passive diffusion? Water, O2 and CO2. How does facilitated diffusion differ from passive diffusion? Facilitated diffusion uses carrier molecules to effectively transport glycerol, sugars, and amino acids. Similarities in passive and facilitated diffusion. Movement of molecules is not energy-dependent. Direction of movement is from high concentration to low concentration. Size of concentration gradient impacts rate of uptake. What is an energy-dependent transport process that uses ATP or proton motive force to move molecules against the gradient? Active transport. Which active transport uses ATP? Primary. Which type of active transport uses PMF or other ion gradient? Secondary, which transport uses a high-energy metabolic pathway. Intermediate, group translocation. What happens in group translocation? Molecules are modified as they are transported across the membrane. The substance between the membrane and the nucleic is called blank. Cytoplasmic matrix. What is the cytoplasmic matrix often packed with? Ribosomes and inclusion bodies. What organism lacks a true cytoskeleton? Mycoplasma bacteria, for example, atypical pneumonia. What are the eukaryotic cell cytoplasmic elements? Microtubules, microfilaments, and intermediate filaments. What type of protein is microtubules made of? Tubulin. What type of protein is microfilaments made of? Actin. Function of MRE-BMBL. Helps determine cell shape, may be involved in chromosome segregation, localizes proteins. An inclusion body found in cyanobacteria and some other aquatic forms helps the organism float on the surface. Gas vacuoles. An inclusion bodies that contain iron in the form of magnetite. Magnetosomes. Types of organic inclusion bodies. Polybihydroxybutyrate glycogen lipid droplets. Type of inorganic inclusion bodies. Polyphosphate or volition granules, sulfur granules. Complex structures consisting of protein and RNA, they are responsible for the synthesis of cellular proteins. Ribosomes. Prokaryotic ribosomes are blank in structure too, but blank than eukaryotic ribosomes. Similar, smaller. What type of ribosomes produce proteins for transport? Bound ribosomes. What is the prokaryotic ribosome called? 70S. What is the eukaryotic ribosome called? 80S. Irregularly shaped region in which the chromosome of the prokaryote is found? Nucleoid. In most prokaryotes, the nucleoid contains a blank chromosome. Single circular. Which organelle is not bounded by a membrane? The nucleoid. Closed circular DNA molecules found in bacteria are called blank. Plasmids. What is not the purpose of the plasmid? Bacterial growth and reproduction. A plasmid that is integrated into the bacterial chromosome is called episome. The loss of plasmid during cell division of bacteria is called blank. Plasmid curing. What is the plasmid capable of conjugation? Fertility plasmid. The plasmid that contains gene that can build resistance against one or several antibiotics or poisons. Resistance plasmid. The plasmid that contains genes coding for collicines, proteins that can kill other bacteria. Plasmids. What is the type plasmid that is able to digest unusual substances, for example, toluene or salicylic acid? Degradative plasmids. The plasmid that can turn a bacterium into a pathogen. Virulence plasmids. What are the functions of the cell wall? Maintain cell shape, counters the effects of osmotic pressure. A barrier that protects cell contents, provides attachment sites for bacteriophages, platform for surface appendages, site of action of some antibiotics. Some important features of a cell wall. Rigid structure, porous, no selectivity. Most important factor in material species differentiation. Cell walls are not identical. The amino acids in peptidoglycan structure only attaches to which sugar? NAM. A macromolecular network of cell wall repeating disaccharide attached by polypeptides to form a lattice. Peptidoglycan. What makes up the disaccharides of the peptidoglycan structure? NAG and NAM. Where is the peptide interbridge more frequently observed? In gram-positive. In gram-positive cell walls, what is linked to the plasma membrane and span peptidoglycan layer? Lipotachoic acids. What is linked to the peptidoglycan in gram-positive cell walls? Wall tachoic acid. Which bacteria cell wall consists of many layers of peptidoglycan? Gram-positive. Consist mainly of alcohol and phosphate found in G+ cell walls. Tachoic acids. What do gram-negative cell walls lack? Tachoic acids. In gram-negative cell walls, the peptidoglycan is bonded to blank. Lipoprotein in the outer membrane. Where is the lipoprotein located in the gram-positive cell walls? In the periplasmic space. What is the area between the outer membrane and the cytoplasmic membrane in the gram-negative cell wall? Periplasmic space. What contains high levels of degradative enzymes and transport proteins in the gram-negative cells? Periplasmic space. Which bacteria cell wall consists of one or very few layers of peptidoglycan and has an outer membrane? Gram-negative. Which cell wall is more complex? A. Gram-positive. B. Gram-negative. Gram-negative. What contributes to the charge of the cell wall? Tachoic acid. In gram-negative cells, what does the lipid bilayer consist of? Lipoproteins, protein, lipopolysaccharides and phospholipids. What does the outer membrane of the gram-negative cell wall serve as? A protective barrier to certain antibiotics, digestive enzymes, heavy metals, and certain dyes. Proteins in the outer membrane of gram-negative cells that allow the passage of selected particles? Porins. What is the most abundant lipoprotein in the outer membrane of gram-negative cell walls? Brown's lipoprotein. What gives the gram-negative cell wall their negative charge? Polysaccharide. What anchors the LPS to the outer membrane in the gram-negative cell walls? Lipid A. Polysaccharide portion composed of sugars in gram-negative cell walls are called blank. Polysaccharides or O antigen. The bottom part of the LPS is called blank. Lipid A. What is the importance of LPS? Protection from host defenses gives the negative charge of the cell, helps stabilize outer membrane structure, can act as an endotoxin. What helps to stabilize the outer membrane structure of the gram-negative cell? Lipid A of the LPS. What part of the LPS in the gram-negative cell can act as an endotoxin? Lipid A. What provides protection from host defenses in the gram-negative cell? O antigen in the LPS. Which is more permeable? A. Plasma membrane. B. Outer membrane. Outer membrane. What causes the outer membrane to be more permeable? The presence of porin proteins and transporter proteins. The middle part of the LPS is called blank. Polysaccharide. Which is closer to Archaea? A. Archaea. B. Bacteria. Archaea. Whose cell wall is more diverse? A. Archaea. B. Bacteria. Archaea. A bacteria without a cell wall is called Mycoplasma. In a bacteria without a cell wall, the plasma membrane is blank than normal. Stronger. What provides the extra strength to the plasma membrane in Mycoplasma? Sterols. True or false? Mycoplasma membrane can change their shape. True. Describe the cell wall of acid-fast bacteria. What are the bacteria stained with an acid-fast staining? Hot stains and with phenol-containing stains. What happens after staining the bacteria in acid-fast staining? The lipid layer in the wall prevents decolorization. What type of bacteria cell wall is poorly permeable, grows slowly and are resistant to chemical disinfectants and lysosome-mediated destruction? Acid-fast. What are archaea closely related to? Eukarya. What feature do archaea and bacteria have in common? Genes for metabolism. What feature do archaea and eukarya have in common? Genes encoding protein. What makes archaea highly diverse? Morphology, physiology, reproduction, and ecology. In what conditions do archaea grow best? Anaerobic, hypersaline pH extremes, and high-temperature habitats. Archaeal shapes. Cocci, rods, branched flat. Archaeal cell walls differ from bacterial cell walls. Like peptidoglycan, most common cell wall is S-layer. In archaea may have protein sheath external to S-layer. S-layer may be outside membrane and separated by pseudomembrane. Pseudomembrane may be outermost layer, similar to gram-positive microorganisms. What is the function of the cytoskeleton? Cell division, protein localization, determination of cell shape. What is the function of FTSC-type tubulin homologs? Cell division. What is the function of MRE-BMBL actin homologs? Maintains cell shape, segregates chromosomes, localizes proteins. Cells are often in what kind of solution? A. Hypertonic, B. Hypotonic, C. Isotonic. Hypotonic. What prevents movement of water into cell, causing swelling and lysis due to osmotic pressure? The cell wall. What does the cell wall not protect against? Plasmolysis. When does plasmolysis occur? When cells are in hypertonic solution. What happens during plasmolysis? Water moves out of cell, causing the cytoplasm to shrivel and pull away from cell wall. What is the importance of plasmolysis? Food preservation, egg-dried foods and jellies. What is the importance of osmotic lysis? The basis of lysosome and penicillin action. Cells that lack a cell wall are called protoplast. What are cells called that have some cell wall remaining? Spheroplast. Where is the periplasmic space located in gram-positive bacteria? Between plasma membrane and cell wall. Where is the periplasmic space located in gram-negative bacteria? Between plasma membrane and outer membrane. Where are periplasmic enzymes found? In the periplasm of gram-negative bacteria. What are some of the functions of periplasmic enzymes? Nutrient acquisition, electron transport, peptidoglycan synthesis, modification of toxic compounds. What type of enzyme is secreted by gram-positive bacteria? Ixoenzymes. What are the functions of ixoenzymes? nutrient acquisition, electron transport, peptidoglycan synthesis, modification of toxic compounds. What are the types of layers of material lying outside the cell wall? Capsules, slime layers, and S layers. What layer outside the cell wall is usually composed of polysaccharides and is well organized and not easily removed from cell? Capsules. What layer outside the cell wall is similar to capsules except diffuse, unorganized, and easily removed? Slime layers. A capsule or slime layer composed of polysaccharides can also be referred to as a blank. Glycolytics. What layer outside the cell wall is regularly structured in layers of protein or glycoprotein and is common among archaea? S layers. What are the functions of capsules, slime layers, and S layers? Protection from host defenses from harsh environmental conditions, viral infections, or predation by bacteria, attachment to surfaces, mobility, protection against osmotic stress. What is required for motility or DNA uptake? Fimbriac pili. What is required for conjugation? Sex pili. Monotrichous. One flagellum. Polar flagellum. Flagellum at end of cell. Amphitrichous. One flagellum at each end of cell. Lophotrichous. Cluster of flagella at one or both ends. Peritrichous flagella. Spread over entire surface of cell. The counterclockwise movement of the flagellum causes the cell to move forward. The clockwise movement of the flagellum causes the cell to move nowhere. Axial filaments cause flexing and spinning movement is called blank. Spirochetes. The movement towards a chemical attractant or away from a chemical repellent is called blank. Chemotaxis. What causes the cell to move toward an attractant? By lowering the frequency of tumbles. What causes the cell to move away from an attractant? By increasing the frequency of runs. What environments are the bacterial endospore resistant to? Heat, radiation, chemicals, desiccation. What is the process of endospore formation called? Sporogenesis or sporulation. What causes an endospore to form? When growth ceases due to the lack of nutrients. What makes an endospore so resistant? Calcium to stabilize the DNA, acid-soluble DNA-binding protein, dehydrated core spore, DNA repair enzymes. The transformation of endospore into a vegetative cell is called germination. Gram-positive cells are stained what color? Purple. What do prokaryotic cells not contain? Sterols. Tuberculosis. Mycobacterium tuberculosis. Atypical pneumonia. Mycoplasma pneumoniae. Leprosy. Mycobacterium leprae. Toxic shock syndrome. Staphylococcus aureus. Opportunist. Pseudomonas aeruginosa. Typhus. Rickettsia prawazici. Most common pneumonia. Staphylococcus pneumoniae. Cholera. Vibrio cholerae. Rocky Mountain fever. Rickettsia rickettsii. Food poisoning. Campylobacter jejuni. What provides a complex environment required for cellular activities? Cytoplasm matrix. What does the cytoplasm of eukaryotes consist of? Liquids, the cytosol, and many organelles. What is a vast network of interconnected filaments within the cytoplasmic matrix? Cytoskeleton. What are the filaments that form the cytoskeleton? Microfilaments, microtubules, intermediate filaments, motor protein. What is the purpose of the cytoskeleton? Plays role in both shape of the cell and cell movement. What is involved in cell motion and shape changes? Microfilaments. What protein is microfilaments made of? Actin. What helps maintain cell shape? Microtubules. What is the purpose of microtubules? Help maintain cell shape. Involved with microfilaments in cell movement, participate in intracellular transport processes. What are the heterogeneous elements of the cytoskeleton? Intermediate filaments. Which cell organelle transports proteins, lipids, and other materials within the cell? ER. What is the major site of cell membrane synthesis? ER. The membranous organelle made of cisternae stacked on each other? Golgi Apparatus. Stacks of cisternae are called dictyosomes. What is the function of the Golgi apparatus? Modification, packaging, secretion of materials. Membrane-bound vesicles are called endosomes. Which organelle contains hydrolytic enzymes needed for digestion of macromolecules? Lysosomes. The uptake of solutes or particles by enclosing them in vesicles or vacuoles pinched off from the plasma membrane? Endocytosis. What are the two types of endocytosis? Phagocytosis and polycytosis. Phagocytosis. The uptake of large particles. Polycytosis. Uptake of small amounts of liquids with its solute molecules. What is the function of lysosomes? Break down materials and waste host defense. Where can proteasomes be observed? Eukaryotes, some bacterium, many archaea. Non-lysosomal protein degradation system. Proteasomes. These ribosomes synthesize non-secretory proteins and non-membrane proteins. Free ribosomes. These are complexes of mRNA with numerous ribosomes. Polyribosomes. The location of enzymes and electron carriers for electron transport and oxidative phosphorylation. Mitochondria. Contains ribosomes, mitochondrial DNA, and large calcium phosphate granules. Matrix. Contains enzymes of the tricarboxylic acid cycle and the oxidation pathway for fatty acids. Matrix. Pigment-containing organelles observed in plants and algae, cytophotosynthetic reactions. Chloroplasts. Site of light reactions. Glycoids. Participates in polysaccharide synthesis in chloroplast. Pyranide. Nuclear envelope. Double membrane structure that delimits nucleus penetrated by nuclear pores. Pores allow materials to be transported into or out of nucleus. What are the reproductive strategies of bacteria and archaea? Haploid only, asexual, binary fission, budding, filamentous. What are the two cycles of the prokaryotic cell cycle? DNA replication and partition, cytokinesis. What is considered the cell cycle of bacteria cells? The replication of the cells. What is the beginning of the cell cycle in bacteria cells? The replication of genetic material. The site at which replication begins. Origin of replication. The site at which replication is terminated. Terminus. Replisome. Group of proteins needed for DNA synthesis. Lag phase. Cell synthesizing new components varies in length. Exponential phase. Also called log phase, rate of growth is constant. Population is most uniform in terms of chemical and physical properties during this phase. In what phase does the total number of viable cells remains constant? Stationary phase. Possible reasons for entry into stationary phase. Nutrient limitation, limited oxygen availability, toxic waste accumulation, critical population density reached. What are the responses to starvation of a cell? Morphological changes, for example, endospore formation, decrease in size, protoplast shrinkage, and nucleoid condensation, production of starvation proteins, long-term survival, increased virulence. Death phase. Cells dying at exponential rate. Death, irreversible loss of ability to reproduce. In some cases, death rate slows due to accumulation of resistant cells. Time required for the population to double in size. Generation. Mean growth rate constant. Number of generations per unit time, usually expressed as generations per hour. What are the two ways one can measure microbial growth? Changes in the number of cells in population. Measure mass of population. What are the two ways of measuring the number of cells? Direct count, viable cell count. What are counting chambers useful for? Counting eukaryotes and prokaryotes. What is the disadvantage of using counting chambers? Gives the count of both living and dead cells. A machine that pumps cells through its opening and counts the number of cells through electron currents. Electron counter. What are electron counters used for? Large microorganisms and blood cells, not prokaryotes. What is the disadvantage of using electron counters? Gives the count of both living and dead cells. Direct count on membrane filters. Cells filtered through special membrane that provides dark background for observing cells. Cells are stained with fluorescent dyes useful for counting. Bacteria with certain dyes can distinguish living from dead cells. What regulates the flow rate of media through vessel to maintain a predetermined turbidity or cell density? The turbidostat. Turbidostat operates best at blank dilution rates. High. Importance of continuous culture methods. What are extremophiles? Organisms that grow under extreme conditions. Organisms that grow over wide ranges of water activity. Osmotolerant organisms. Organisms that require high levels of NaCl to grow. Halophiles. The negative logarithm of the hydrogen ion concentration. Telephone. Organisms that can grow in pH levels between 0 and 5.5. Acidophils. Organisms that can grow in pH levels between 5.5 and 7. Neutrophils. Organisms that can grow in pH levels between 8.5 and 11.5. Alkalophiles. Most acidophils and alkalophiles maintain an internal pH near blank. Neutral. The optimum temperature is close to the blank temperature. Maximum. What is the distance between the minimum and maximum temperatures? 30 degrees. Psychrophiles. Organisms that can grow in extremely cold temperatures. Psychotrophs. Organisms that can grow in cold temperatures. Mesophiles. Organisms that grow in warm temperatures. Thermophiles. Organisms that grow in hot temperatures. Hyperthermophiles. Organisms that grow in extremely high temperatures. What type of organisms have more saturated fatty acids? Hyperthermophiles. What type of organisms have more unsaturated fatty acids? Psychrophiles. Organisms that need oxygen. Obligate aerobe. Organisms that prefer oxygen. Facultative anaerobe. Organisms that don't need oxygen but can still grow in the presence of oxygen. Aerotolerant anaerobe. Organisms that cannot grow in oxygen. Strict anaerobe. Organisms that require a less concentration of oxygen that is present in the atmosphere. Microorophile. What are the two enzymes produced by obligate aerobe and facultative anaerobe to protect them from toxic compounds? Superoxide dismutase catalase. Which organism produces only SOD? Aerotolerant anaerobe. Which organism does not produce SOD or catalase? Strict anaerobe. Which organism produces SOD and low levels of catalase? Microorophile. Barotolerant organisms. Adversely affected by increased pressure but not as severely as non-tolerant organisms. Barophilic organisms. Require or grow more rapidly in the presence of increased pressure. What are the effects of ionizing radiation on certain organisms? Mutation greater than death disrupts the chemical structure of many molecules, including DNA, X, gamma and X-ray. What are the effects of UV radiation on organisms? Mutation greater than death causes formation of thymine dimers in DNA. DNA damage can be repaired by two mechanisms: photoreactivation, dimers split in presence of light; dark reactivation, dimers excised and replaced in absence of light. What are the effects of visible light on organisms? At high intensities generate singlet oxygen. What protects light-exposed microorganisms from photoxidation? Carotenoid pigments. Non-cultivable vegetative prokaryotes. Microorganisms that can temporarily lose their ability to grow using normal cultivation methods. Microbial communication and cooperation. Quorum sensing. How do microorganisms communicate? By secreting and detecting chemical signals. What chemical is produced by many gram-negative cells for communication? Acetylmosylserine lactone. Process of quorum sensing. AL or other signal molecules diffuse across plasma membrane at high concentrations. It enters the cell. Once inside the cell, it induces expression of target genes that regulate a variety of functions. What is needed to grow, transport, and store microorganisms in the lab? Culture media. Defined or synthetic media. All components and their concentration of media are known. Complex media. Contains some ingredients of unknown composition and or their concentration. Media components. Peptone extract agar. Supportive or general media. Support the growth of many microorganisms. X. TSA. selective media. Favor the growth of some microorganisms and inhibit the growth of others. XMNTSA. Differential media. Distinguish between different groups of microorganisms based on their biological characteristics. Pure culture. A population of cells arising from a single cell. Techniques used to isolate pure culture. Streak plate, pour plate, spread plate. The destruction or removal of all viable organisms. Sterilization. The killing, inhibition, or removal of pathogenic organisms. Disinfection. The reduction of microbial population to levels deemed safe. Sanitization. The prevention of infection of living tissue by microorganisms. Antisepsis. Chemical agents that kill or inhibit the growth of microorganisms when applied to the tissue. Antiseptics. Agents that kill microorganisms or inhibit their growth. Antimicrobial agents. What type of antimicrobial agents kill? Cytal agents. What type of antimicrobial agents inhibit growth? Static agents. Germicide. Kills pathogens and many non-pathogens but not necessarily endospores. Suffix indicating that agent kills. Side X bactericides, fungicides, algicides and viricides. When are microorganisms considered dead? When they are unable to reproduce in conditions that normally support the reproduction. What are the factors that influence the killing of microorganisms? Population size, population composition, concentration or intensity of an antimicrobial agent, duration of exposure, temperature, local environment. What are physical methods of killing microorganisms? Heat, low temperatures, filtration, radiation. Which heat-killing method is most effective against all types of microorganisms? Moist heat. How is moist heat effective in killing microorganisms? Degrades nucleic acids, denatures proteins, and disrupts membranes. Which heat-killing method is less effective? Dry heat. Thermal death time. The shortest time needed to kill all microorganisms in a suspension at a specific temperature and under defined conditions. Time required to kill 90% of microorganisms or spores in a sample at a specific temperature. Decimal reduction time. What is an autoclave used for? To kill endospores efficiently, uses saturated steam under pressure to reach temperatures above boiling. What is pasteurization? Reduces total microbial population and thereby increases the shelf life of treated material. Flash pasteurization. 72 degrees Celsius for 15 seconds, then rapid cooling. Ultra-high temperature sterilization. 140 to 150 degrees Celsius for 1 to 3 seconds. Reduces microbial population or sterilizes solutions of heat-sensitive materials by removing microorganisms and is also used to reduce microbial populations in air. Filtration. What is used as a skin antiseptic? Iodine. Commonly used as laboratories and hospitals disinfectants. Phenolics. At high concentration, what can kill spores? Iodine. Iodine complexed with organic matter. Iodophore. Important in disinfection of water supplies and swimming pools, used in dairy and food industries, effective household disinfectant. Chlorine. Chemical that destroys vegetative bacteria and fungi but not spores. Chlorine. can react with organic matter to form carcinogenic compounds. Chlorine, used to inactive some viruses. Alcohols. What chemical is used to denature proteins and possibly dissolve membrane lipids? Alcohols. Examples of heavy metals. Ions of mercury, silver, arsenic, zinc, and copper. Combines with and inactivate proteins may also precipitate proteins. Heavy metals. Detergents. Organic molecules with hydrophilic and hydrophobic ends act as wetting agents and emulsifiers. Highly reactive molecules. Aldehydes. Sporicidal and can be used as chemical sterilants. Aldehyde. Combines with and inactivate nucleic acids and proteins. Aldehydes. Ethylene oxide and beta-propiolactone are both. Sterilizing gases. Used to sterilize heat-sensitive materials. Sterilizing gases. Kills microbes and spores. Sterilizing gases. Phenol coefficient test. The potency of a disinfectant is compared to that of phenol. What organisms are used for the phenolic test? Salmonella typhi and Staphylococcus aureus. Used to test conditions that approximate normal use of disinfectant. In-use testing determines rate at which selected bacteria are destroyed by various chemical agents. Dilution test. Chemical agents used to treat disease. Chemotherapeutic agents. Destroys pathogenic microbes or inhibit their growth within host. Chemotherapeutic agents. Paul Ehrlich. Developed concept of selective toxicity, identified dyes that effectively treated African sleeping sickness. Who identified arsenic compounds that effectively treated syphilis? Sahashiro Hado. Gerhard Domagk, Jacques and Therese Truffaut. Discovered sulfonamides and sulfa drugs. Who was the first to discover penicillin? Ernest Duchenne. Who accidentally discovered penicillin? Alexander Fleming. Who discovered the effectiveness of penicillin? Florey, Chain, and Heatley. An antibiotic active against tuberculosis. Streptomycin. Salman Waxman. Awarded Nobel Prize for the discovery of streptomycin. The ability of a drug to kill or inhibit pathogen while damaging host as little as possible. Selective toxicity. The drug level required for clinical treatment. Therapeutic dose. The drug level at which the drug becomes too toxic for the patient. Toxic dose. The ratio of toxic dose to therapeutic dose. Therapeutic index. The bacteria species that produces most of the antibiotics. Streptomyces spp. 2 ways in determining the level of antimicrobial activity. Minimal inhibitory concentration, minimal lethal concentration. Involves inoculating media containing different concentrations of drugs. Dilution susceptibility tests. Broth or agar with the lowest concentration showing no growth. Minimum inhibitory concentration. Broth from which microbes can't be recovered. MLC. How does one measure antimicrobial sensitivity? Disk diffusion tests. Disk diffusion test. Discs impregnated with specific drugs are placed on agar plates inoculated with test microbe drug diffuses from disc into agar, establishing concentration gradient. Observe clear zones around discs. Kerb-Bauer method. Standardized method for carrying out disc diffusion tests. Sensitivity and resistance determined using tables that relate zone diameter to degree of microbial resistance. Table values plotted and used to determine if concentration of drug reached in body will be effective. The E-test. Uses strips instead of discs. Intersection of elliptical zone of inhibition with strip indicates MIC. What can be used to determine the concentration of a drug in blood? Microbiological, chemical, immunological, enzymatic, or chromatographic assays. Concentration of drug at infection site must be blank MIC to be effective. A less than B greater than C equal to greater than. What can impact the pathogen by targeting some function necessary for its reproduction or survival? Antimicrobial agents. Sulfonamides. Inhibit folic acid synthesis. Broad spectrum. What are the factors influencing the effectiveness of antimicrobial drugs? Ability of drug to reach site of infection. Susceptibility of pathogen to drug. Ability of drug to reach concentrations in body that exceed MIC of pathogen. The ability of a drug to reach the site of the infection depends on what? Mode of administration. For example, oral, topical, non-oral routes. What is the most crucial feature of the penicillin molecule? Beta-lactam ring. Most penicillins are blank derivatives. 6-aminopenicillinic. What do most penicillin-resistant organisms produce? Beta-lactamase, which hydrolyzes a bond in this ring. Penicillin mode of action. Blocks the enzyme that catalyzes transpeptidation. Prevents the synthesis of complete cell walls, leading to lysis of cell. Acts only on growing bacteria that are synthesizing new peptidoglycan. Cephalosporins. Structurally and functionally similar to penicillins. Broad-spectrum antibiotics that can be used by most patients that are allergic to penicillin. What are the two glycopeptide antibiotics? Vancomycin and teicoplanin. What two glycopeptide drugs inhibit cell wall synthesis? Vancomycin and teicoplanin. Important for treatment of antibiotic-resistant staphylococcal and enterococcal infections. Vancomycin. What was previously considered the drug of last resort? Vancomycin. Where do protein synthesis inhibitors bind to on the bacterial ribosome? 30S or 50S ribosomal subunit. Which penicillin must be taken by shot and not by mouth because it is destroyed by stomach acid? Penicillin G. What are the inhibition cell wall synthesis drugs? Penicillin, cephalosporins, vancomycin and bacitracin. Nucleic acid synthesis inhibition. What is an example of a drug that does nucleic acid synthesis inhibition? Rifampin. What are examples of drugs for protein synthesis inhibition? Streptomycin, tetracycline, chloramphenicol, and erythromycin. Mechanisms of drug resistance. Exclusion of drug. The drug can bind to or penetrate pathogen. Pump drug out. Inactivation of the drug. Chemical modification of drug by a pathogen. Alteration of target enzyme or organelle. Use of alternative pathways or increased production of the target metabolite. The blank is a measure of disinfectant efficiency in which the disinfectant being evaluated is compared to phenol. Phenol coefficient test. The blank is the length of time it takes for a population of microorganisms to double in number. Generation double time. Macroscopically visible growths of clusters and microorganisms on solid media are called a blank colony. Many bacteria facilitate the uptake of iron by secreting low molecular weight molecules called blank to form complexes with the iron that can then be readily transported into the cell. Siderophores. The type used in a laminar flow biological safety cabinet is called a blank filter. HEPA. An agent that kills bacteria is referred to as blank. Bactericides. Organisms that can grow in habitats with low water availability by maintaining a high internal salt concentration. Osmotolerant. Blank are required organic compounds because they are essential cell components or precursors of such components that cannot be synthesized by the organism. Growth factors. Growth media that will support the growth of many different types of microorganisms are called blank growth media, whereas those that are supplemented by blood or other nutrient sources in order to support the growth of fastidious organisms are called blank media. General purpose enriched. Colonies grow most rapidly at the blank where oxygen and nutrients are readily available. However, they grow less rapidly at the blank where these materials have been depleted. Edges, center. Test organisms for phenol coefficient test are A blank B blank. A Salmonella typhi, B Staphylococcus aureus. N80 stands for blank. Nicotinamide adenine dinucleotide. A molecule that binds to an enzyme at a location other than the active site and thereby alters the enzyme shape, making it inactive or less active, is a blank inhibitor. Non-competitive. When a chemotherapeutic agent is effective against many different pathogens, it is said to be a blank antibiotic. Broad spectrum. Botulism is caused by an organism called blank. Clostridium botulinum. In the F case of the plating techniques, bacterial populations are expressed as blank. Colony-forming units. FAD stands for blank. Flavin adenine dinucleotide. A molecule that binds non-covalently to an enzyme at the active site and thereby prevents a substrate from binding and reacting is a blank inhibitor. Competitive. The blank is the substrate concentration needed for an enzyme to achieve maximal velocity. KM. Plasmids that carry genes responsible for antibiotic resistance are called blank. R plasmid resistance plasmid. NADP stands for blank. Nicotinamide adenine dinucleotide phosphate. Blank is a continuous culture system where an essential nutrient is in limiting quantities. Chemostat. Organisms that grow well between 40–80 C are called blank. Thermophiles. Nutrients required in large quantities are called blank. Macronutrients. Killing, inhibition, or removal of pathogenic organisms is called blank. Disinfection. The molecules formed by an enzyme-catalyzed reaction are called blank. Product. Blank is the lowest concentration of drug that kills pathogen. Minimal lethal concentration. A process in which molecules are chemically modified and simultaneously transported into the cell is called blank. Group translocation. Bacteria that can survive well at high temperature usually have high concentration of blank fatty acid in their plasma membrane. Saturated. Randomness or disorder of a system is called blank. Entropy. An agent that prevents the growth of bacteria without causing irreversible damage to the bacteria is referred to as blank. Bacteriostatic. A reaction in which the forward rate is equal to the reserve rate is said to be at blank equilibrium. An agent that kills fungi is referred to as blank fungicide. Disruption of an enzyme structure with loss of activity caused by extremes of pH, temperature, or other factors is called blank denaturation. Bacteria that can survive well at very low temperatures usually have high concentration of blank fatty acid in their plasma membrane. Unsaturated. Blank radiation is an effective means of sterilization but is limited only to surface. UV. Blank is an antibiotic that blocks cell wall synthesis in bacteria. Penicillin. Transport of two different substances can be linked. If the transport is in the same direction, it is called blank. If the transport is in opposite directions, it is called blank. Subport, antiport. True or false? Water activity is inversely proportional to osmotic pressure. True. Organisms that can use carbon dioxide as their sole or principal source of carbon are autotrophs. A culture in a closed vessel to which no additional medium is added and from which no waste products are removed is called a blank culture. Batch. The prevention of infection caused by microorganisms is called antisepsis. The killing or removal of all viable organisms is called sterilization. The killing of vegetative forms of pathogenic microorganisms on inanimate objects is called disinfection. Which of the following is not a form of ionization radiation? A. Ultraviolet radiation at 260 nanometers. B. X radiation. C. Gamma radiation. D. All of these are forms of ionizing radiation. Ultraviolet radiation at 260 nanometers. T or F. Heavy metals are effective antimicrobial agents but are not widely used because of their high toxicity to humans. True. An open system in which the growth rate is maintained by the removal and addition of media at such a rate as to maintain a constant cell density is called a turbidostat. An open system in which the growth rate is maintained by adding a nutrient at the same rate that medium containing microorganisms is removed is called chemostat. Differential. Selective and differential. T or F. During the lag phase of microbial growth, the cells are metabolically inactive. False. Which of the following is considered a cardinal growth temperature? A. The minimum temperature. B. The maximum temperature. C. The optimum temperature. D. All of these are considered cardinal growth temperatures. All. Organisms that do not require oxygen for growth but grow better in its presence are called facultative anaerobes. Nutrients can be concentrated from dilute solutions by active transport and group translocation. Which of the following is most effective against resistant endospores? A. Autoclaving. B. Glutaraldehyde. C. Pasteurization. D. All of these are very effective against endospores. Autoclaving. T or F. Alcohols are widely used as antiseptic and disinfectants because they are effective against endospores as well as vegetative cells. False. T or F. When a young, vigorously growing culture of bacteria is transformed to fresh medium of the same composition, the lag phase is usually short or absent. True. T or F. When a young, vigorously growing culture of bacteria is transformed to fresh medium of a different composition, the lag phase is usually short or absent. False. Iodine can be complexed with an organic carrier to from water-soluble, stable complex is called blank, which release iodine slowly and eliminate skin burns and irritation associated with iodine use. Iodophores. Organisms that obtain energy from the oxidation of either inorganic or organic compounds are called chemotrophs. Organisms that obtain electrons from the oxidation of organic compounds are called organotrophs. When comparing dry heat and moist heat sterilization, dry heat is slower. Which of these methods can be used to determine the number of viable organisms in a sample? A. Light scattering in a spectrophotometer. B. Measuring total cell mass. C. Measuring colony-forming units per ml. D. Counting in known volume of cells in a hemocytometer. C. Which of the following cannot be used to estimate the viable count of microorganisms in a culture? A. Direct counts of microbial cells. B. Measurements of microbial biomass. C. Counts of viable cells using colony growth procedures. D. None of the above. E. Only A and B. E. Organisms that obtain electrons from the oxidation of inorganic compounds are called lithotrophs. A culture system with constant environmental conditions maintained through continual provision of nutrient and removal of waste called blank culture system. Continuous. T or F. Ultraviolet radiation is generally more harmful than ionizing radiation. False. T or F. One of the major advantages of a continuous culture system is that the cells can be maintained in the exponential growth phase for extended periods of time. True. C. The length of the lag phase can vary depending on A. The condition of the microorganisms. B. The nature of the growth medium. C. Both A and B are correct. D. Neither A nor B are incorrect. C. Organisms that ignore oxygen and grow equally well in its presence or absence are called aerotolerant. T or F. Ethylene oxide can be used to sterilize materials in sealed plastic wrap because it can penetrate the wrapping material. True. Microbial populations are reduced by blank when exposed to a lethal agent. A constant fraction. Organisms that have a narrow range of growth temperatures are said to be stenothermal. In order to determine the minimal lethal concentration, the culture is first inoculated into the medium containing the drug and then subcultured into medium containing no drug. In order to be effective, the concentration of a drug in the body must be higher than the pathogen's minimal inhibitory concentration. Which of the following can be used to isolate pure cultures of bacteria from mixtures? A. Spread plates. B. Streak plates. C. Pour plates. D. All of these. D. The increased temperature needed to reduce the decimal reduction time to 10% of its value is the Z-value. Which of the following influence the efficiency of an antimicrobial agent? A. Concentration of the agent. B. Duration of exposure. C. Temperature. D. All of these. E. Two of the choices. D. T or F. Penicillin is frequently injected but not normally given orally because it is rapidly degraded in the stomach. True. Organisms that grow well at 0 degrees and have optimum growth temperatures of 15 degrees or lower are called psychrophiles. The movement of molecules from an area of high concentration to an area of low concentration by random thermal agitation is called passive diffusion. The movement of molecules from an area of high concentration to an area of low concentration with the use of a carrier molecule embedded in the membrane is called facilitated diffusion. Sterilization is accomplished by blank all viable microorganisms. A. Killing. B. Removing. C. Either killing or removing. D. Neither killing nor removing. C. Given a log-phase bacterial culture with 2.0 x 10 to the 6 cells per ml and a generation time of 40 minutes, how long does it take the culture to reach a density of 1.6 x 10 to the 7 cells per ml? 2 hours. Given a log-phase bacterial culture with 10 to the 6 cells per ml and a generation time of 30 minutes, how long does it take the culture to reach a density of 1.6 x 10 to the 7 cells per ml? 2 hours. Given a log-phase bacterial culture with 10 to the 6 cells per ml and a generation time of 45 minutes, how long does it take the culture to reach a density of 6.4 x 10 to the 7 cells per ml? 4.5 hours. Which of the following is a characteristic of active transport? A. Saturable uptake. B. Use of ATP or proton motive force as a source of metabolic energy. C. Can move materials against a concentration gradient. D. All of these are characteristics of active transport. All. Which of these is an alkylating agent that is used to sterilize the plastic tubing of heart-lung machines? A. Phenol. B. Mercaptophenol. C. Ethylene oxide. D. 70% ethanol. Ethylene oxide. T or F. Phosphorus can be obtained from organic sources only. False. Cells may enter stationary phase because of A. the depletion of an essential nutrient. B. A. lack of available oxygen. C. the accumulation of toxic waste products. D. all of the choices. All. Agar is an excellent gelling agent for microbiological media because it is not degraded by most microorganisms, and solid agar remains solid until the temperature is raised to 100 degrees Celsius, and liquid agar remains liquid if the temperature is lowered to 45 degrees Celsius. Which of the following can be most useful to isolate pure cultures of oxygen-sensitive bacteria from mixtures? A. Spread plates. B. Streak plates. C. Pour plates. D. all of the choices. Pour plates. The membranes of psychrophilic bacteria have relatively high levels of blank fatty acids, which allow them to remain semi-fluid at cold temperatures. Unsaturated. The membranes of thermophiles have high levels of blank fatty acids, which allow them to remain semi-fluid at cold temperatures. Saturated. All. Organisms that grow in mud under relatively non-turbulent bodies of water are likely to be anaerobes. T or F. The primary mechanism by which DNA is damaged by ultraviolet radiation involves the formation of thymine dimers. True. T or F. Ultraviolet radiation is an effective means of sterilizing surfaces. True. T or F. Aerobic organisms do produce protective enzymes called superoxide dismutase and catalase. True. Which of the following environmental factors generally do not have an impact on the efficiency of an antimicrobial agent? A. Penthouse. B. Viscosity. C. Concentration of organic matter. D. Refractive index. D. D. Which of the following heavy metals are still used in some cases as germicides? A. Lead acetate. B. Copper sulfate. C. Silver nitrate. D. Mercury chloride. E. Only B and C are correct. E. Although heavy metals are no longer widely used as germicides, blank is an effective algicide in lakes and swimming pools. Copper sulfate. Which of the following inhibits bacterial growth but does not kill bacteria? A. Lysozyme. B. Bactericidal agent. C. Bacteriostatic agent. D. Antiseptic agent. C. Which kind of sterilization procedure takes advantage of the size of microorganisms? A. Autoclaving. B. Open flame. C. Use of chemicals. D. Filtration. D. T or F. The rate of killing by an antimicrobial agent may slow when the microbial population has been greatly reduced because the remaining population may have a higher proportion of resistant organisms. True. The standard reduction potential of a redox reaction is a measure of the tendency of the blank-to-blank electrons. Reactant lose. T or F. The effects of most antimicrobial drugs are greater if the microorganisms are actively growing. True. A non-protein component of an enzyme that is loosely attached to the protein component is referred to as a coenzyme. A chemical reaction that requires an input of energy in order to proceed is blank. Endergonic. Which of the following is used by microorganisms to become resistant to a particular drug? A. Enzymatic inactivation of the drug. B. Exclusion of the drug from the cell. C. An alternate metabolic pathway that bypasses the drug-sensitive step. D. All of the choices. All. B. Which of the following is likely to have the most toxic side effects to humans? A. Inhibitors of cell wall synthesis. B. Inhibitors of protein synthesis. C. Disruptors of cell membrane structure. D. Inhibitors of DNA synthesis. C. T or F. A drug that disrupts a microbial function not found in animal cells usually has a higher therapeutic index. True. T or F. A drug that disrupts a microbial function not found in animal cells usually has a higher therapeutic index. True. T or F. Gracefulvin is used to treat systemic mycosis. False. T or F. Gracefulvin is used to treat superficial mycosis. True. Which of the following antibiotic is broad-spectrum? A. Penicillin. B. Ampicillin. C. Streptomycin. D. Vancomycin. E. Two of these. E. The light reactions of photosynthesis which form ATP and NADPH take place in the thylakoid membrane. T or F. Permease proteins that aid in the transport of nutrients resemble enzymes in their specificity for the substance to be transported. Each carrier is selective and will transport only a closely related set of substances. True. Which statement is true about prokaryotes? A. They contain nucleus. B. They lack ribosomes. C. They do not divide by mitosis. D. They usually lack a cell wall. E. All contain a single circular DNA molecule as genetic material. C. 97. Which of the following processes do not show saturable uptake rates? A. Active transport. B. Passive diffusion. C. Facilitated diffusion. D. Neither passive diffusion nor facilitated diffusion show saturable uptake rates. B. Which of the following is useful in distinguishing between prokaryotic and eukaryotic cells? A. The use of DNA for the storage of genetic information. B. The presence of ribosomes for the synthesis of proteins. C. The presence of membrane-delimited organelles within the cytoplasm. D. All of these. C. The most selective antibacterial agents are those that interfere with bacterial cell wall synthesis. This is because bacterial cell walls have a unique structure not found in eukaryotic cells. The proteasome A functions to degrade most eukaryotic proteins. B. Produces peptides for antigen presentation during immunological responses. C. Hydrolyzes ubiquitin-marked eukaryotic proteins in an ATP-dependent process. D. All of the above are true. D. A blank is a special type of lysosome used by a cell to digest portions of its own cytoplasm. A. Suicide vacuole. B. Turnover lysosome. C. Recycling vacuole. D. Autophagic vacuole. Autophagic vacuole. Which of the following is a function of pasteurization? A. Killing pathogenic microorganisms. B. Reducing the total microbial population. C. Increasing the shelf life of the product. D. All of these are functions of pasteurization. D. The process of heating milk products to 72 C for 15 seconds in order to reduce the microbial population is called blank pasteurization. Flash. C. The blank is the electron donor in a redox reaction. Reductant. The blank is the electron acceptor in a redox reaction. Oxidant. Which of the following is used as an electron carrier by living organisms? A. NAD plus. B. NADP plus. C. Ubiquinone. D. All of these are used as electron carriers by living organisms. D. Competitive inhibition can be overcome by adding excess substrate. If all available enzyme molecules are binding substrate and converting it to product as rapidly as possible, the reaction is said to be proceeding at blank velocity. Maximal. When the end product of a pathway inhibits catalysis of the first step of that pathway, this phenomenon is called feedback inhibition. T or F. The Michaelis constant of an enzyme is the substrate concentration that produces maximum velocity. False. T or F. The Michaelis constant of an enzyme is the substrate concentration that produces half velocity. True. The drug level required for the clinical treatment of a particular infection is called the therapeutic dose. T or F. Treatment of fungal infections is more difficult than treatment of bacterial infections because the greater metabolic similarity between the fungi and their host limits the ability of a drug to have selective toxicity. True. T or F. When the amount of enzyme present is held constant, the rate of a reaction will continue to increase as long as the substrate concentration increases. False. T or F. Enzymes increase the rate of a reaction by increasing molecular motion, thereby providing kinetic energy to drive the reaction. False. Which of the following is normally treated by topical application of antifungal drugs? A. Systemic mycosis. B. Superficial mycosis. C. Subcutaneous mycosis. D. Intramuscular mycosis. B. Which of the following is not a method of heat sterilization? A. Autoclaving. B. Dendalization. C. Pasteurization. D. All of these are methods of heat sterilization. C. Moist heat sterilizes by denaturing protein. Which of the following is a desirable general characteristic of antimicrobial drugs? A. Selective toxicity. B. Broad spectrum of activity. C. Must be bactericidal, not bacteriostatic. D. All of the above. D. T or F. The trace amounts of micronutrients needed by microorganisms can usually be supplied in water and regular media components. True. Which of the following contributes to resistance to killing? A. Formation existence of endospores. B. Increased age of culture. C. Inherent genetic differences among organisms. D. All of these may contribute to resistance to killing. D. Which will require a longer time to kill? A larger population of microorganisms. B. A smaller population of microorganisms. C. Killing will be equally as rapid in a large or a small microbial population. D. There is no way to predict which will require a longer time to kill. A. Organic molecules that serve as wetting agents and emulsifiers and can be used as disinfectants if they are cationic are called detergents. T or F. Dry heat methods usually require lower temperatures and shorter exposure times than moist heat methods to achieve the same degree of killing because of the drying effects of this form of heat. False. T or F. Dry heat methods usually require higher temperatures and longer exposure times than moist heat methods to achieve the same degree of killing because of the drying effects of this form of heat. True. T or F. Germicides kill all pathogenic organisms and endospores. False. Which of the following is a function of the mitochondrion? A. Carboxylic acid cycle enzyme reactions. B. Electron transport. C. ATP synthesis. D. All of these are functions of the mitochondrion. D. The exponential growth rate of a bacterial culture is affected by all of the following except A. The genetic potential of the organism. B. The composition of the growth medium. C. The number of cells initially inoculated into the culture vessel. D. The condition of incubation. C. Which of the following is a sterilizing agent that can be used on heat-sensitive materials? A. Ethylene oxide. B. Beta-propiolactone. C. Gamma radi. All of the above. D. D. Enzyme activity can be controlled by allosteric regulation, B covalent modification, C feedback inhibition, D all of these, E only A and C. D. T or F. The equilibrium constant for a redox reaction is called the standard reduction potential. True. Genes for drug resistance are usually located A on the chromosome, B on plasmid, C on mitochondrial DNA, D both A and B, E both B and C. D. When antiseptics and disinfectants are compared, antiseptics are generally A. less toxic, B. more toxic, C. equally as toxic, D. unpredictable in toxicity. Less toxic. Which of the following is not a method of heat sterilization? A. autoclaving, B. incineration, C. pasteurization, D. heating in a dry air oven. Pasteurization. Moist heat sterilization at 100 C kills all of the following except A. bacterial endospores, B. fungal spores, C. viruses, D. bacterial endospores and fungal spores. Bacteria endospores. Gamma radiation. A is a type of non-ionizing radiation, B has poor penetrating power, C is used to sterilize some food products, D has a longer wavelength than UV radiation. C. The first product to be commercially treated by pasteurization was A. milk, B. wine, C. beer, D. cheese. Wine. Pasteurization is used to A. kill any pathogens present, B. retard spoilage, C. sterilize beverages, D. kill any pathogens present and retard spoilage. D. Which of the following is a function of pasteurization? A. Killing pathogenic microorganisms, B. reducing the total microbial population, C. increasing the shelf life of the product, D. all of the choices. D. B. The two most important alcohol germicides are blank and blank. A. ethanol, methanol, B. ethanol, isopropanol, C. methanol, butanol, D. methanol, isopropanol. B. Disinfectants are generally regulated by the A. Food and Drug Administration, B. Environmental Protection Agency, C. the National Institutes of Health, D. Centers for Disease Control and Prevention. B. Antiseptics are generally regulated by the A. Food and Drug Administration, B. Environmental Protection Agency, C. the National Institutes of Health, D. Centers for Disease Control and Prevention. A. Anthrax, Bacillus anthracis, Listeria monocytogenes, Listeriosis. What is the development and spread of drug-resistant pathogens? Superinfection. How to prevent emergence of drug resistance? Give drug in high concentrations. Give 2 or more drugs at same time. Use drugs only when necessary. Possible future solutions. What drug is used to prevent influenza infections by blocking penetration and uncoats the virus? Amantadine. What drug inhibits herpes virus enzymes involved in DNA and RNA synthesis and function? Adenine arabinoside. What drug inhibits herpes virus and cytomegalovirus DNA polymerase? Foscarnet. What drug inhibits herpes virus DNA polymerase? Acyclovir. What is a broad-spectrum anti-DNA drug for viruses? Cefovir. What drug reverses the nucleotide transcription for HIV? Azithromycin or zidovudine. What is the viral protease inhibitor for HIV? Ritonavir. Reverse transcription inhibitors for viruses? Azithromycin or zidovudine, lamivudine. Protease inhibitors for viruses? Ritonavir. What is metabolism? The total of all chemical reactions in the cell and is divided into 2 parts. What are the two types of metabolism? Anabolism and catabolism. Catabolism. Fueling reactions. Energy-conserving reactions provide ready source or reducing powder generate precursors for biosynthesis. Anabolism. The synthesis of complex organic molecules from simpler ones requires energy from fueling reactions. What are the four types of work carried out by organisms? Energy, chemical work, transport work, mechanical work. What is energy? Capacity to do work or to cause particular changes. What is chemical work? Synthesis of complex molecules. What is transport work? Take-up of nutrients, elimination of wastes, and maintenance of ion balances. What is mechanical work? Movement of organisms or cells and movement of internal structures. What is used to transfer energy from the cell's energy-conserving systems to the systems that carry out cellular work? ATP. What is the first law of thermodynamics? Energy cannot be created or destroyed. It can be changed from one form to another. Chemical energy in food is converted to chemical energy in ATP and then converted to mechanical energy of muscle contraction. This is an example of what? The first law of thermodynamics. What is the second law of thermodynamics? Energy cannot be changed from one form into another without a loss of usable energy. 25% of chemical energy of gasoline is converted to move a car. Rest is lost as heat. This is related to which law of thermodynamics? The second law. When muscles convert chemical energy in ATP to mechanical energy, some is lost as heat. This is an example of what law of thermodynamics? The second law. What is entropy? Measure of randomness or disorder. Is the entropy of the universe always decreasing or increasing? Increasing. What is free energy? The amount of energy that is free to do work after a chemical reaction. The change in free energy is noted as what symbol? Delta G. What does negative delta G represent? Products have less free energy than reactants. The reaction occurs spontaneously. What reactions have a negative delta G and energy is released? Exergonic reactions, spontaneous reactions. What reactions have a positive delta G and energy is gained? Endergonic reactions. What are reactions that have a free energy difference near zero? Reversible reactions. Blank have great specificity for the reaction catalyzed and the molecules acted on. Protein catalysts. Blank is a substance that increases the rate of a reaction without being permanently altered. Catalysts. What are reacting molecules? Substrates. Substances formed by reactions. Products. What is a coenzyme? Protein component of an enzyme. Non-protein component of an enzyme is called cofactor. How do enzymes lower the activation energy? By increasing concentrations of substrates at active site of enzyme by orienting substrates properly with respect to each other in order to form the transition state complex. As the concentration of substrates increase, the rate of the reaction blank increases. What happens to the enzyme structure and activity when the temperature and pH rises too much above optima? Denaturation. Directly competes with binding of substrate to active site. Competitive inhibitor. binds enzyme at site other than active site, changes enzyme shape so that it becomes less active. Non-competitive inhibitor. Who discovered that some RNA molecules also catalyze reactions? Thomas Seck and Sidney Altman. What is the importance of regulating metabolism? Conservation of energy and materials. What are the three major mechanisms for regulating metabolism? Metabolic channeling, regulation of the synthesis of a particular enzyme, direct stimulation or inhibition of the activity of a critical enzyme. What is metabolic channeling? Differential localization of enzymes and metabolites. What is feedback inhibition? Inhibition of one or more critical enzymes in a pathway that regulates entire pathway. Where can the ED pathway be found? E. coli and Pseudomonas. What is the net gain of ATP in the glycolysis cycle? 2. What is the net gain of ATP in the ED pathway? 1. What is the net gain of NADH in ED pathway? 1. In what pathway is 5C sugar made? Oxidative decarboxylation. What is the final electron acceptor in aerobic respiration? Oxygen. What is the final electron acceptor in anaerobic respiration? Different exogenous acceptors or organic acceptors. As electrons pass through the electron transport chain to the final electron acceptor, a blank is generated and used to synthesize ATP. Protein motive force. Chemoorganic fueling processes use what type of electron acceptor? Endogenous. What process does not involve the use of an electron transport chain nor the generation of a proton motive force? Chemoorganic fueling process is fermentation. How is ATP synthesized in chemoorganic fueling processes? Only by substrate-level phosphorylation. Overview of aerobic catabolism. Large molecules equals greater than small molecules. Initial oxidation and degradation to pyruvate. Oxidation and degradation of pyruvate by the tricarboxylic acid cycle. What are the two functions of organic energy sources? Oxidize to release energy, supply carbon and building blocks for anabolism. What are amphibolic pathways? Functions both as catabolic and anabolic pathways. What are the three common routes to break down glucose to pyruvate? Glycolysis, pentose phosphate pathway and Naudorff pathway. Where does the glycolytic pathway occur? In the cytoplasmic matrix of both prokaryotes and eukaryotes. What is an example of an amphibolic pathway? The pentose phosphate pathway. Kerb cycle. What is the source of electrons for the pentose phosphate pathway? The NADH produced. Which cycle completes oxidation and degradation of glucose and other molecules? Kerb cycle. During the pentose phosphate pathway, how is ATP produced? No ATP is produced. NADH is converted into NADH. In what organisms is the Kerb cycle common? Aerobic bacteria, free-living protozoa, most algae and fungi. For each acetyl-CoA molecule oxidized, the TCA cycle generates 2 molecules of CO2, 3 molecules of NADH, 1521 GTP. What is oxidized in the electron transport chain? Most ATP made from NADH and Fad2. What do reducing agents do in redox reactions? Donate electrons. What do oxidizing agents do in redox reactions? Electron Acceptor. A measurement of the tendency of reducing agents to lose electrons. Standard reduction potential. Is pyruvate reduced or oxidized during fermentation? Reduced. What reduces pyruvate during fermentation? NADH. What does NADH reduce pyruvate to during fermentation? Lactic acid. What is the electron acceptor in fermentation? Pyruvate or pyruvate derivate. Can amino acids be fermented? Yes. In the electron transport and oxidative phosphorylation pathways, how is ATP produced? When NADH and Fat2 are oxidized. What enzyme is used to hydrolyze fatty acids to glycerol? Lipase. What is produced during the glycolytic pathway from 2-NADH? 5-ATP and 2-ATP. Can bacteria use other carbohydrates than glucose? Yes. How many ATP are produced in the Krebs cycle? 5 ATP. How many ATP are produced in the tricarbon acid cycle at substrate level phosphorylation? 2 ATP. How many ATP are produced in the tricarbon acid cycle during oxidative phosphorylation with 6-NADH? 15 ATP. How many ATP are produced in the tricarbon acid cycle during oxidative phosphorylation with 2-Fat2? 3 ATP. In the electron transport chain, electrons flow from carriers with a more blank charge to carriers with a more blank charge. Negative, positive. What is the released energy from the electron transport chain used to make? ATP by oxidation phosphorylation. Where is the prokaryotic ETCs located? Plasma membrane. What is the glycolytic pathway also called? Embden-Meyer's pathway. What is the pentose phosphate pathway also called? Hexose monophosphate pathway. Where are prokaryotic etc. located? Plasma membrane. How do prokaryotic etc. differ from mitochondrial etc.? Different electron carriers may be branched, may be shorter, may have lower PO ratio. What is the most widely accepted explanation of oxidative phosphorylation? Chemotaxis hypothesis. Energy released during electron transport is used to establish a proton gradient and charge difference across the membrane. This is called blank blank. Proton motive force. The enzyme that uses proton movement down gradient to catalyze ATP synthesis. ATP synthase. T or F aerobic respiration provides much more ATP than fermentation. True. The reduction of nitrate to nitrogen gas is called blank. Denitrification. What causes loss of soil fertility? Denitrification. An energy-yielding process in which indigenous organic molecules serve as both electron donors and acceptors. Fermentation. In alcoholic fermentation, what is produced as a result of the reduction of pyruvate? ATO and CO2. What is used as an energy source in the absence of external nutrients? Reserve polymers at glycogen starch PHB. Enzyme that hydrolyzes protein to amino acids. Protease. The removal of an amino group from amino acid. Deamination. What is the result of deamination? Resulting organic acids converted to pyruvate acetyl-CoA or TCA cycle intermediate. Oxidation of inorganic molecules are carried out by blank. Chemolithotrophs. In sulfur-oxidizing bacteria, ATP can be synthesized by blank. Both oxidative phosphorylation and substrate-level phosphorylation. T or F chemolithotrophs may switch from chemolithotrophic metabolism to chemorganotrophic metabolism. True. T or F chemolithotrophs may switch from autotrophic metabolism to heterotrophic metabolism. True. What is photosynthesis? Energy from light trapped and converted to chemical energy. Reactions in which light energy is trapped and converted to chemical energy. Light reaction. Reactions in which the energy captured in the light reactions is used to reduce the O2 and synthesize cell constituents. Dark reactions. What reaction is a Calvin cycle? Dark reaction. Where did the dark reaction take place? In the stroma of chloroplast of eukaryotic organisms. In prokaryotic organism it takes place in the cytoplasm. The energy captured in ATP and NADH during the light reaction is used for what? To power the second phase of photosynthesis, carbon fixation. What is the first reaction of the Calvin cycle? 3-ribulose 1,5-bisphosphate plus 6CO2 greater than 6,3-phosphoglycerate. In cyanobacteria organic photosynthesis, what is produced? O2. What eukaryotic organisms do oxygenic photosynthesis? Algae and plants. In photosynthesis, what is the electron donor? Water. What is the final product of oxidation in photosynthesis? Oxygen. What is the major pigment in photosynthesis? Chlorophyll. What are the products of light reactions? ATP and NADH. In non-oxygenic photosynthesis, what is the major pigment? Bacteria chlorophyll. How is ATP produced in non-oxygenic photosynthesis? ATP is produced by chemismosis using PMF. This process is also called photophosphorylation. What enzyme catalyzes the Calvin cycle? Rubisco. What is a virus? A cellular infectious agent. T or F viruses can be used as a new source of therapy. True. T or F viruses are important members of the aquatic world. True. Why are viruses important to the aquatic world? Move organic matter from particulate to dissolved. Why are viruses important in evolution? Transfer genes between bacteria, others. What is an important model in molecular biology? Viruses. A complete virus particle. Virion. Consists of one or more molecules of DNA or RNA enclosed in coat of protein. Virion. T or F virions cannot reproduce independent of living cells nor carry out cell division but can exist extracellularly. True. What are bacterial viruses called? Bacteriophages. What type of cells do viruses affect most? Eukaryotic. Viruses are classified into families based on genome structure, life cycle, morphology, genetic relatedness. What are hosts for bacteriophages? Usually cultivated in broth or agar cultures of suitable, young, actively growing bacteria. Membrane structure surrounding some viruses. Viral envelopes. What is the largest animal virus? Pox virus. What are the steps in viral multiplication? Attachment to host cell entry, uncoating of genome, synthesis, assembly, release. The phage life cycle that culminates with host cell bursting, releasing virions. Lytic cycle. Phages that lyse their host during the reproductive cycle / virulent phages. Specific surface structures on the host to which viruses attach. Receptor sites. Protects phage DNA from host restriction. Endonucleases, enzymes that cleave DNA at specific sequences. HMC glucosylation. Proteins that aid in the construction of procapsid. Scaffolding proteins. Nolytic relationship between a phage and its host. Lysogeny. Integrated phage genome. Prophage. Infected bacterial host. Lysogens. Lysogenic conversion. Change in host phenotype induced by lysogeny. Examples of an organism that undergo lysogenic conversion through the modification of its lipopolysaccharide structure. Salmonella. Examples of an organism that undergo lysogenic conversion through the production of toxin. Corynebacterium dipylori. How do naked viruses release virions? By lysis of host cells. How do envelope viruses release virions? Formation of envelope and release usually occur concurrently. Lysosome damage causes the release of blank enzymes into cell. Hydrolytic. What is the result in the alteration of the plasma membrane in host cells? Can lead to attack of host cell by immune system, can lead to cell fusion forming syncytium. What are the mechanisms of host cell damage and cell death? Inhibition of host DNA, RNA, and protein synthesis, lysosome damage, alteration of plasma membrane, toxicity from high concentration of viral proteins, formation of inclusion bodies, chromosomal disruptions, transformation of host cell into malignant cell. Types of persistent infections. Chronic virus infections, latent infection. What type of viruses are almost always detectable? Chronic. What type of viruses stop reproducing and remains dormant for some time? Latent infections. What are the possible causes of persistent infections? Integration of viral genome into host genome, decrease in antigenicity of virus, mutation to less virulent and slower reproducing form. Types or viruses and cancer. Tumor, neoplasia, anaplasia, metastasis. Abnormal new cell growth and reproduction due to loss of regulation. Neoplasia. Reversion to a more primitive or less differentiated state. Anaplasia. Spread of cancerous cells throughout body. Metastasis. Cancer-causing genes. Oncogenes. Possible mechanisms by which viruses cause cancer. Carry oncogene into cell and insert it into host genome, altered cell regulation due to changes in kinase activity or production of regulatory proteins, insertion of promoter or enhancer at next to cellular oncogene. Infectious agents composed only of circular ssRNA. Viroids. Infectious agent thought to be composed only of protein. Prions. Causes disease in plants. Viroids. Proteins are catabolized by hydrolytic cleavage to amino acids by the action of enzymes called blank. Protease. Microscopic or macroscopic degenerative changes or abnormalities in the infected host cells and tissues are called blank. Cytopathic effect. Icosahedral viruses are constructed from ring or knob-shaped units called blank capsomers. In glycolytic pathway, the conversion of glucose to glucose 6-phosphate is catalyzed by an enzyme called blank glucokinase. The hypothesis that proton motive force drives ATP synthesis is called the blank chemosmosis. Blank are glycoprotein spikes protruding from the outer surface of viral envelope. Peplimers. Disaccharides and polysaccharides can be processed as nutrients after first being cleaved to monosaccharides by either blank or blank hydrolases, phosphorylases. The synthesis of ATP from ADP and Pi when coupled with the exergonic enzymatic breakdown of a high-energy molecule is called blank phosphorylation. Substrate level. A process that involves the use of pyruvate and or pyruvate derivatives as electron acceptors is called blank fermentation. Lysis of host brought by several T4 proteins such as blank and blank. Endolysin, Nolan. Change in host phenotype induced by lysogeny is called blank Lysogenic Conversion. Decrease in rate of sugar metabolism when microbes shifted from anaerobic to aerobic conditions is known as blank Pasteur effect. The most commonly accepted hypothesis for the production of ATP that results from electron transport system is called the blank hypothesis. Chemotaxis. Blank is used as the terminal electron acceptor for oxidizing NADH and NAD in aerobic respiration, but molecules other than oxygen are used as the terminal electron acceptors in blank oxygen anaerobic. When mitochondria pass electrons donated by NADH through the electron transport chain to molecular oxygen, a theoretical maximum of blank molecules of ATP can be made oxygen molecule reduced. 3. The basic proteins that associate with and condense eukaryote DNA are called blank histones. Viruses that are polyhedrons with 20 sides are said to have blank symmetry. Icosahedral. Blank are glycoprotein spikes protruding from the outer surface of the viral envelope. Peplimer. Localized areas of destruction occurring on plants that have been infected by a virus are referred to as blank lesions. Nucleoid. Simple molecules are further broken down in cells in a process called blank, during which energy stored in their chemical bonds is used to powder the production of ATP. Catabolism. For further derivation of energy, aerobic cells must convert pyruvate into acetyl coenzyme A by stripping off a CO2 molecule. This process is known as blank oxidative carbylation. Plants, algae and cyanobacteria use a two-stage type blank in the light-dependent reactions. Photosystem. Non-capsid proteins that aid in the assembly of virion structures but do not become incorporated into the virion are referred to as blank proteins. Scaffolding. A bacterium carrying a dormant phage inserted in the chromosome is called a blank lysogen. Blank and blank are two enzymes very important in pentose phosphate cycle to combine various sugars. Transferase, transaldolase. Major photosynthetic pigment in purple sulfur bacteria is blank bacterial chlorophyll. Blank is an enzyme that splits fructose 1,6-bisphosphate to 2,3-carbon containing molecules in glycolysis. Aldolase or 1,6-biphosphate aldolase. The phage genome inserted in the chromosome of bacteria is called a blank prophage. An enzyme that can join molecules using ATP energy can be called blank ligase. In glycolysis phosphate cycle 3-phosphogluconate is decarboxylated to a 5C sugar called blank ribulose. All of the reactions of glucose oxidation that follow glycolysis involving the transfer of electrons to their final acceptor, oxygen, take place in eukaryotic cells in the blank mitochondria. Fats undergo a process called blank oxidation in which the products are acetyl coenzyme molecules. Beta oxidation. Even though the Calvin cycle reactions require the products of the light reactions, its reactions can occur in the blank stroma. Organisms that directly reduce almost all of their pyruvate to lactate are called blank homolactic. Disruption of an enzyme structure with loss of activity caused by extremes of pH, temperature, or other factors is called blank denaturation. A complex formed during a reaction that resembles both the substrates and the products is called the blank state complex. Transition. The molecules formed by an enzyme-catalyzed reaction are called blank product. In allosteric regulation, the effector molecule that binds to the site on the enzyme is called blank kinase. T or F oxygen is required for the regeneration of NAD from NADH. False. When a viral genome is integrated into the host chromosome but does not result in the lysis of the host cell, it is referred to as atemperate infection, B. avirulent infection, C. lysogeny-dialytic infection. Lysogeny. Which of the following is not true of viroids? A. Viroids are small circular ssRNA molecules. B. Viroids have no protein capsids. C. Viroids do infect plants and some prokaryotic cells. D. All of these are true of viroids. C. Viroids do infect plants and some prokaryotic cells. Which of the following is not true of viroids? A. Viroids are small circular ssRNA molecules. B. Viroids have no protein capsids. C. Viroid RNA does not serve as mRNA nor does it direct the synthesis of mRNA. D. All of these are true of viroids. D. All of these are true of viroids. Which of the following is not true of viroids? A. Viroids are small circular ssRNA molecules. B. Viroids have no protein capsids. C. Viroid RNA does not serve as mRNA nor does it direct the synthesis of mRNA. D. Extracellular viroids have a lipid bilayer envelope. D. Extracellular viroids have a lipid bilayer envelope. Which of the following can serve as phage receptor sites? A. Lipopolysaccharides and tachoic acids. B. Pili. C. Membrane proteins. D. All of these can serve as phage receptor sites. D. All of these can serve as phage receptor sites. T or F. The presence or absence of an envelope is not useful in classifying viruses because any given virus may at one time have an envelope and at another time not have an envelope. False. What do the M and ED pathways have in common? A. They're considered types of glycolytic pathways. B. They require oxygen. C. They are found only in obligate anaerobes. D. All of the above. A. They're considered types of glycolytic pathways. What do the M and ED pathways have in common? A. They're considered types of glycolytic pathways. B. They require oxygen. C. They are found only in obligate anaerobes. D. Both can be found in some bacteria. E. Two of the above. A. They're considered types of glycolytic pathways. T or F. Metabolic pathways can be either catabolic or anabolic but not both. False. C. They produce defective interfering particles. T or F. Both ED and M pathways are very common in bacteria. False. T or F. Parvoviruses are the simplest known animal viruses. True. Which of the following is a function of late viral proteins? A. Formation of the virion capsid. B. Assembly of mature virions by non-capsid proteins. C. Lysis of host cells. D. All of these are functions of late viral proteins. D. All of these are functions of late viral proteins. T or F. The Voges Proskera test is positive for butylenediol fermenters but negative for mixed acid fermenters. True. Which events are part of TCA cycle? A. Citrate reacts with oxalostate to form acetyl-CoA. B. Glucose is converted to fructose. C. NAD is regenerated. D. Carbon dioxide, reduced coenzymes and ATP are produced. D. Carbon dioxide, reduced coenzymes and ATP are produced. T or F. Metabolism of fats produces more ATP molecules per gram than metabolism of carbohydrates or proteins. True. B. It may have the normal bases found in prokaryotic and eukaryotic DNA, or it may have one or more unusual bases. The number of ATP molecules generated per atom of oxygen that is reduced electrons are passed from NADH or reduced alpha-D to D. Oxygen utilization ratio. B. Energy yield. C. PO ratio. D. Phosphorylation coefficient. PO ratio. Which liberates the most energy in the formation of ATP? A. Alcohol fermentation. B. Sugar fermentation. C. Aerobic respiration. D. Anaerobic respiration. C. Aerobic respiration. Viral capsid protein subunits are called A-imers. B. Elastomers. C. Protomers. D. Viromers. C. Protomers. Which of the following is true about viral envelopes? A. The envelope proteins are virus-specific. B. The envelope lipids and carbohydrates are derived from the host. C. Both of these answers are true. D. Neither of these answers are true. C. Both of these answers are true. What occurs when a pyruvate molecule proceeds toward and through the Krebs cycle? A. H atom end up in NADH and Fat2 plus 3 carbon dioxides are formed. B. Pyruvate converted to heat energy. C. Fatty acids are synthesized. D. Carbon dioxide combine with acetyl-CoA and form oxalostate. A. H atom end up in NADH and Fat2 plus 3 carbon dioxides are formed. The synthesis of ATP from ADP and Pi when coupled with the exergonic enzymatic breakdown of a high-energy molecule is called blank phosphorylation. A. Chemostatic. B. Oxidative. C. Substrate level. D. Conformational change. Substrate level. C. Both protect the viral genetic material and aid in the transfer of the viral genetic material between host cells. In the TCA cycle, 2 carbons are removed from citric acid in the form of blank, thereby regenerating oxalostate to complete the cycle. Acetyl-CoA. B. Ethanol. C. Carbon dioxide. D. Methanol. Carbon dioxide. T or F. The most efficient pathway for ATP production in animal cells is by glycolysis. False. C. Both specific surface proteins on the bacteriophage and specific receptor proteins on the host cell. Amino acids are processed first by the removal of the amino group through A. Deamination. B. Transamination. C. Either deamination or transamination. D. Neither deamination or transamination. C. Either deamination or transamination. C. Both energy production and provision of carbon skeletons for biosynthesis of cell components. The major function of the TCA cycle is A. Energy production. B. Provision of carbon skeletons for biosynthesis of cell components. C. Direct oxidation of 4 and 5 carbon sugars during anaerobic growth. D. Energy production and provision of carbon skeletons for biosynthesis of cell components. D. Energy production and provision of carbon skeletons for biosynthesis of cell components. 30. During breakdown of fatty acids, carbons are removed blank at a time as blank. A1. Carbon dioxide. B1. Methane. C2. Acetyl-CoA. D2. Ethanol. C2. Acetyl-CoA. The most common type of molecule functioning as an animal virus receptor is A lipoprotein, B glycoprotein, C phosphoprotein, D tachoic acid. Glycoprotein. T or F, the injection mechanism used by bacteriophages whereby the nucleic acid penetrates the cell leaving the protein coat outside, is not used by any known animal viruses. False. A blank genome exists as several separate non-identical molecules that may be packaged together or separately. A diploid, B segmented, C polyploid, D fractionated. Segmented. Viruses with single-stranded RNA as their genome for which the base sequence is the same as the viral mRNA are said to be blank viruses. A plus-stranded, B minus-stranded, C mRNA-like, D none of these. Plus-stranded. In which of the following stages of the viral infectious cycle do enveloped viruses usually acquire their envelopes? A penetration, B component biosynthesis, C assembly, D release. Release. Which of the following is not true regarding ATP synthesis? A. They require proton motive force to make ATP. B. They span the inner membrane of mitochondria. C. The proton flow is outward during ATP synthesis. D. The subunits of ATP synthase undergo conformational changes during ATP. The proton flow is outward during ATP synthesis. Which of the following statements concerning the proton motive force is correct? A. This takes place only in bacteria. B. It reduces organic carbon compounds. C. PMF is based on the differential concentration of the proton across the membrane. D. All of the above. PMF is based on the differential concentration of the proton across the membrane. Which of the following statements concerning the proton motive force is correct? A. This takes place only in bacterial cells. B. It reduces organic carbon compounds. C. PMF is based on the differential concentration of the proton across the membrane. D. A, B, and C are correct. PMF is based on the differential concentration of the proton across the membrane. What takes place when pyruvate is converted to acetyl-CoA? A. One molecule of carbon dioxide is given off. B. One molecule of Fad2 is produced from FADC. The conversion takes place in the cytoplasm. D. All of the above. E. Only A and C are correct. Only A and C are correct. What takes place when pyruvate is converted to acetyl-CoA? A. One molecule of carbon dioxide is given off. B. One molecule of Fad2 is produced from FADC. The conversion takes place in the cytosol. D. None of these. 1 CO2. T or F. During glycolysis, one molecule is converted to one molecule of pyruvate. False. In the absence of oxygen, eukaryotic cells are restricted to H. Amosmotic phosphorylation, B. Cyclic photophosphorylation, C. Non-cyclic photophosphorylation, D. Oxidative phosphorylation, E. Substrate level phosphorylation. Substrate level phosphorylation. The decarboxylation of pyruvate produces ANADH, B. Acetyl-CoA, C. CO2, D. ATP, E. Only A, B, and C are correct. Only A, B, and C are correct. The electron transport chain, a series of membrane-associated electron carriers, loses most of the energy by driving several transmembrane A proton pumps, B electron pumps, C sodium, potassium pumps, D active transport pumps, E water pumps. Proton pumps. The major function of the TCA cycle is A. Energy production, B. Provision of carbon skeletons for biosynthesis of cell components, C. Direct oxidation of 4 and 5 carbon sugars during anaerobic growth, D. Energy production and provisions of carbon skeletons for biosynthesis of cell components. Energy production and provisions of carbon skeletons for biosynthesis of cell components. Which of the following is not true of viruses? A. Viruses are acellular. B. Viruses consist of one or more molecules of DNA or RNA enclosed in a coat of protein and sometimes in other more complex layers. C. Viruses are obligate intracellular parasites. D. Viruses replicate by binary fission. Viruses replicate by binary fission. Which of the following is not true of viruses? A. Viruses are acellular. B. Viruses consist of one or more molecules of DNA or RNA enclosed in a coat of protein and sometimes in other more complex layers. C. Viruses are obligate intracellular parasites. D. All of these are true of viruses. All of these are true of viruses. The nucleic acids carried by viruses usually consist of A, DNA, B, RNA, C, either DNA or RNA but not both simultaneously. D, both DNA and RNA simultaneously. Either DNA or RNA but not both simultaneously. It may have the normal bases found in prokaryotic and eukaryotic DNA, or it may have one or more unusual bases. Most important character in viral classification is A. Nucleic acid type, B. Nucleic acid strandedness, C. Presence or absence of an envelope, D. All of the choices. Nucleic acid type. Viruses are subgrouped according to A. Nucleic acid type, B. Nucleic acid strandedness, C. Presence or absence of an envelope, D. All of the choices. All. Viruses with single-stranded RNA as their genome for which the base sequence is the same as the viral mRNA are said to be blank viruses. A plus stranded, B minus stranded, C mRNA like, D none of these. Plus stranded. T or F. The presence or absence of an envelope is not useful in classifying viruses because any given virus may at one time have an envelope and at another time not have an envelope. False. Which of the following is a function of late viral proteins? A. Formation of the virion capsid, B. Assembly of mature virions by non-capsid proteins, C. Lysis of host cells, D. All of the choices. All. Fatty acids are metabolized by the blank pathway. A. Alpha oxidation, B. Beta oxidation, C. Gamma oxidation, D. Delta oxidation. Beta oxidation. Production of ATP using the energy liberated by the electron transport system is generally referred to as blank phosphorylation. A. Chemotaxis, B. Oxidative, C. Substrate level deconformational change. Oxidative. Production of ATP using the energy liberated when electrons from reduced chemical bonds are passed through the electron transport system is generally referred to as blank. A. Chemotaxis, B. Respiration, C. Substrate level deconformational change. Chemotaxis. If all available enzyme molecules are binding substrate and converting it to product as rapidly as possible, the reaction is said to be proceeding at blank velocity. A. Terminal, B. Maximal, C. Optimal, D. Infinite. Maximal. T or F. Oxygen is required for the regeneration of NAD from NADH. False. Which of the following can be used as electron acceptors during anaerobic respiration? A. Oxygen, B. Lactate, C. Ethanol, D. Sulfate. Sulfate. The protein coat surrounding the viral genome is called the A capsule, B capsid, C matrix, D envelope. Capsid. The breakdown of larger, more complex molecules into smaller, simpler ones with the release and trapping of some energy contained within those molecules is called A. Anabolism, B. Catabolism, C. Metabolism, D. None of these. Catabolism. Both protect the genetic material and in the transfer of the genetic material between host cells. What occurs when a pyruvate molecule proceeds toward and through the Krebs cycle? A. H atom end up in NADH and Fat2 plus 3CO2 are formed. B. Pyruvate converted to heat energy. C. Fatty acids are synthesized. D. Carbon dioxide combine with acetyl-CoA and form oxalostate. H atom end up in NADH and Fat2 plus 3CO2 are formed. General classes of enzymes found in cells include A. Lyase, B. Metallase, C. Elosterase, D. Fat. Lyase. If an enzyme consists of a protein component and a non-protein component, the protein component is referred as the A apoenzyme, B. coenzyme, C. haloenzyme, D. prosthetic group. Apoenzyme. In the TCA cycle, 2 carbons in the form of blank are added to oxalostate at the start of the cycle. Acetyl-CoA B ethanol C carbon dioxide D methanol. Acetyl-CoA T or F. Like bacteria and eukaryotic microorganisms, viruses can be cultured on artificial media. False. T or F. When the amount of enzyme present is held constant, the rate of a reaction will continue to increase as long as the substrate concentration increases. False. What do the M and ED pathways have in common? A. They're considered types of Glycolytic Pathway. B. They require oxygen. C. They're found only in obligate anaerobes. D. All of the above. They're considered types of Glycolytic Pathway. A complete enzyme that consists of a protein component and a non-protein component is called A. Apoenzyme. B. Heteroenzyme. C. Holoenzyme. D. Multi-enzyme. Holoenzyme. T or F. Metabolic pathways can be either catabolic or anabolic but not both. False. Which of the following is true about viral envelopes? A. The envelope proteins are virus-specific. B. The envelope lipids and carbohydrates are derived from the host. C. Both of these answers are true. D. Neither of these answers is true. Both. Enzyme activity can be controlled by allosteric regulation. B. Covalent modification. C. Feedback inhibition. D. All of these. All. T or F. An organism may use glycolysis and the pentose phosphate pathway simultaneously. True. In higher eukaryotes, most aerobically generated ATP comes from a glycolysis. B. The TCA cycle. C. The electron transport chain. D. All of these generate equal amounts of ATP. The electron transport chain. If the non-protein component of an enzyme is loosely attached to the protein, it is called A. Apoenzyme. B. Coenzyme. C. Holoenzyme. D. Prosthetic group. Coenzyme. If the non-protein component of an enzyme is firmly attached to the protein, it is called A. Apoenzyme. B. Coenzyme. C. Holoenzyme. D. Prosthetic group. Prosthetic group. T or F. Bacterial viruses are so named because they have the same prokaryotic cell structure as their bacterial hosts. False. T or F. Usually the last step in a metabolic pathway is a pacemaker reaction that is catalyzed by a regulatory enzyme. False. Enzymes do not show saturation effect. A. C. The nucleic acids carried by viruses usually consist of A, DNA, B, RNA, C, either DNA or RNA but not both simultaneously. D. Both DNA and RNA simultaneously. C. What occurs during fermentation? A. Membrane-bound system contains cytochromes to produce ATP. B. Fermentation regenerates NAD as pyruvate is converted to ethanol or lactate. C. Acetyl-CoA is regenerated. D. Citric acid is produced. B. Which of the following is not used as an electron carrier by living organisms? A. NAD plus B. NADP plus C. Fad. D. ATPE. All of these are used as electron carriers by living organisms. ATP. T or F. The Michaelis constant of an enzyme is the substrate concentration that produces maximum velocity. False. In which of the following stages of the viral infectious cycle do enveloped viruses usually acquire their envelopes? A. Penetration. B. Component biosynthesis. C. Assembly. D. Release. Release. Which of the following is mismatched? A. Anabolism biosynthesis. B. Catabolism buildup of organic compounds. C. Glycolysis breakdown of sugar into pyruvate. D. Plant phototrophs. B. When a rival genome is integrated into the host chromosome but does not result in the lysis of the host cell, it is referred to as a lysogeny. B. Avirulent infection. C. Tempered infection. D. Lytic infection. A. What happens to the oxygen that is used in cellular respiration? A. It is oxidized to from water. B. It is lost. C. It is produced to form water. D. It is converted to carbon dioxide. C. The blank is the electron acceptor in a redox reaction. A. Oxidant. B. Reductant. C. Enzyme. D. Product. A. The blank is the electron donor in a redox reaction. A. Oxidant. B. Reductant. C. Enzyme. D. Product. B. T or F. Some endergonic reactions can be made to proceed forward if they are coupled to hydrolysis of one or more of the phosphates of ATP. True. D. How is ATP produced during respiratory metabolism? A. By glycolysis. B. By the Krebs cycle. C. By photophosphorylation. D. A, B, and C. E, A, and B. E. How is ATP produced during the Embden-Meyerf pathway? A. Substrate-level phosphorylation. B. Fermentation. C. Cyclic phosphorylation. D. Oxidative phosphorylation. A. The standard reduction potential of a redox reaction is a measure of the tendency of the blank-to-blank electrons. A. Reductant, gain. B. Reductant, lose. C. Oxidant, gain. D. Oxidant, lose. B. D. Which test is used to detect a mixed acid fermentation such as E. coli? A. Catalase. B. Voges proscar. C. Simmons citrate. D. Indole E. methyl red. E. In a one-step growth experiment, the early period in which no infective virions are found even inside the infected host cells is called the blank period. A. Latent. B. Eclipse. C. Rise. D. Plateau. B. T or F. An organism does not use glycolysis and the pentose phosphate pathway simultaneously. False. One of the most important coenzymes that accepts electrons hydrogens is A. NAD plus B. NADH. C. ATPD. NADPH. Ribosome. A. The ability of an enzyme to catalyze a reaction can be affected by chemical and physical factors such as A. Temperature. B. Pentose. C. Salt concentration. D. 2 of the above. E. All of the above. All. A. A. An electron carrier that is used in harvesting energy from glucose molecules in a series of gradual steps in the cytoplasm is A. Pyruvate. B. Cyclic AMP. C. ATPD. NAD plus E. NADH. D. In the absence of oxygen, hydrogen atoms generated by glycolysis and donated to organic molecules in a process called A. Fermentation. B. Decarboxylation. C. Chemosmosis. D. Electron transport chain reactions. E. Acetyl-CoA formation. A. In oxidative respiration, energy is harvested from glucose molecules in a sequence of 4 major pathways. Which one of the following is not one of these 4 pathways? A. Krebs cycle. B. Glycolysis. C. Electron transfer through the transport chain. D. Beta oxidation. E. Pyruvate oxidation. D. The decarboxylation of pyruvate produces A. NADH. B. Acetyl-CoA. C. CO2. D. ATPE. Only A, B, and C are correct. E. B. B. The enzymes of the Krebs cycle in prokaryotic cell are located in the A. cytoplasm. B. Intermembrane space of mitochondria. C. Periplasm. D. Outer membrane of the mitochondria. E. Matrix of the mitochondria. A. E. In the dark reactions of photosynthesis, CO2 is added to a 5-carbon sugar phosphate known as acyclic AMP-BNADH-CNAD plus D-RuBPE-Chem. D. Which of the following is common to both cellular respiration and the light reactions of photosynthesis? A. The transfer of electrons to glucose. B. The chemismotic formation of ATP. C. Oxygen is one of the byproducts. D. Mitochondria are essential organelles. E. Must have light. B. Sulfur bacteria produce ATP in a photosynthetic process called A. The Calvin cycle. B. The Krebs cycle. C. Glycolysis. D. Cyclic photophosphorylation. E. Non-cyclic photophosphorylation. D. Photosystem A differs from photosystem II in that the following is not made directly from the process: A. ATP. B. NADH. C. NADPH. D. Carbohydrates. E. Water. C. Which part of the chloroplasts contain the Calvin cycle enzymes? A. Stroma. B. Thylakoids. C. Grana. D. Envelope. Christae. A. What products of light reactions of photosynthesis are used in the Calvin cycle? A. Oxygen and protons. B. Carbon dioxide and water. C. ATP and NADPH. D. ADP and NADP. E. Glucose and oxygen. C. C. Keneal used sulfur bacteria in his research on photosynthesis. His equation involved carbon dioxide and hydrogen sulfide. The bacteria produce sulfur in green plants, which molecule is split to form the oxygen. A. Water. B. Carbon dioxide. C. Glucose. D. Chlorophyll. Water. What type of cell respiration occurs when an organic molecule accepts electrons? A. Aerobic respiration. B. Anaerobic respiration. C. Fermentation. D. Catabolism. E. Digestion. B. B. Who was the first person to propose that the cause of tobacco mosaic disease is an infectious agent different from bacteria because it could pass through a porcelain filter that retains all bacteria? A. Jenner. B. Ivanowski. C. Beechrink. D. Pasteur. C. A blank essay is most useful for determining the viability of a viral preparation. A. Direct count. B. Hemagglutination. C. plaque. D. PCR. C. The two major types of symmetry found in viruses are A. Icosahedral and radial. B. Icosahedral and helical. C. Helical and radial. D. Radial and bilateral. B. T or F. The largest of the viruses are similar in size to some small bacteria and are large enough to be seen with a light microscope. True. In order for bacteriophage to be released from the host by lysis mechanism, enzymes are required to A. halt ATP synthesis, B. stabilize the cytoplasmic membrane and cell wall, C. damage the cell wall and cytoplasmic membrane, D. degrade the host chromosome. C. Entry of a virus into the lytic cycle after lysogeny has been established is called A. Lysogenic conversion. B. Lysogenic reversion. C. Induction. D. None of the choices. C. T or F. Glucosylation of hydroxymethylcytosine reduce protects phage T4 DNA from cleavage by bacterial restriction enzymes. True. Which of the following morphologies are found among the bacteriophage? A. Tailless phage with icosahedral symmetry. B. Tailed phage with binal symmetry. C. Filamentous phage with helical symmetry. D. All of these are found in a bacteriophage. D. Some viruses do not have a capsid. B. Nucleic acid. C. Protomer. D. Envelope. D. Glycoprotein spikes protruding from the outer surface of viral envelopes may be involved in A. Viral recognition of host cells. B. Viral attachment of host cells. C. Both viral recognition of and viral attachment to host cells. D. Neither viral recognition of nor viral attachment to host cells. C. Which of the following may be used as sources of energy by chemolithotrophs? A. Hydrogen gas. B. Ammonia. C. Elemental sulfur. D. All of these. D. In the process of non-cyclic photophosphorylation, A. Water is split to form oxygen gas. B. Electrons from water flow through electron transport chains resulting in synthesis of ATP and NADH. C. Photosystems I and II or both require D. Two of the above are true. E. A, B, and C are true. E. In the process of non-cyclic photophosphorylation, A. Water is split to form oxygen gas. B. Electrons from water flow through electron transport chains resulting in synthesis of ATP and NADH. C. Photosystems I and II or both require D. All of the above are true. D. B. In aerobic conditions, it takes blank sugar to produce the same amount of ATP when compared to anaerobic conditions. A. More B. Let's see the same amount of less. T or F enveloped viruses may enter their host cells by fusion of their envelope with the cytoplasmic membrane, thereby depositing their nucleocapsid within the cell. True. T or F all prokaryotic photosynthesizers are anexogenic. False. T or F all eukaryotic photosynthesizers are oxygenic. True. T or F reduction of nitrate to nitrite by paracoccus is an example of anaerobic respiration. True. T or F reduction of nitrate to nitrite by paracoccus is an example of aerobic respiration. False. B. T or F a few viruses are as large as smallest bacteria. True. Methods useful for purification of viruses include A. Electron microscopy, B. Differential gradient centrifugation, C. Hemagglutination, D. X-ray diffraction. B. Mitochondrial electron transport takes place A. on the outer mitochondrial membrane, B. on the inner mitochondrial membrane, C. in the fluid matrix of the mitochondria, D. both the inner and on the outer mitochondrial membrane. B. A chemical reaction that requires an input of energy in order to proceed is blank. A. Exergonic, B. Endergonic, C. Impossible, D. None of these. B. B. Which of the following is used by microorganisms to become resistant to a particular drug? A. Enzymatic inactivation of the drug, B. Exclusion of the drug from the cell, C. An alternate metabolic pathway that bypasses the drug-sensitive step, D. All of these. D. Photosynthetic electron transport in cyanobacteria takes place A. on the plasma membrane, B. in the chloroplast, C. in the cytoplasm, D. on the thylakoid membranes. A. T or F bacterial electron transport chains may be comprised of fewer components and have lower PO ratios than mitochondrial transport chains. True. The most common pathway for conversion of glucose to pyruvate is A. Nerdautoff, B. pentose phosphate, C. Embden-Meyerhoff, D. A and C. C. The largest viruses known are the A. herpesviruses, B. hepatinoviruses, C. adenoviruses, D. poxviruses. D. Viroids are A. proteins that may infect man and other animals causing spongiform encephalopathy, B. a class of so-called slow viruses, C. a new class of small single-stranded DNA viruses that infect some animals, D. short infectious single-stranded RNAs that can infect some plants. D. C. Blank are able to degrade infecting bacteriophage DNA, thus protecting the host cell. A. Protection endonucleases, B. Protection exonucleases, C. Restriction endonucleases, D. Restriction exonucleases. C. Hemagglutination is A. the clumping together of red blood cells in the presence of a viral suspension, B. the binding of iron in the process of a viral suspension, C. the clumping together of infected cells in the presence of a viral suspension, D. none of these. A. Enzymes found in virus particles, A. may be required for viral attachment to host cells, B. may be involved in viral entry into host cells, C. may be involved in the replication of viral nucleic acid, D. all of these. All. The pentose phosphate pathway provides AATP, BNADP, HC4 or 5 carbon sugars for amino acid and nucleic acid synthesis, D. all of these. All. In an enveloped virus, the part of the virus including the nucleic acid genome and the surrounding protein coat but not the envelope is called the A capsid, B nucleocapsid, C matrix, D virion. B. T or F in viral infections, all of the viral genes are usually expressed prior to the replication of viral nucleic acid. False. T or F. The electron transport systems in eukaryotes and prokaryotes use different electron carriers. True. Most ATP production in aerobic bacteria is via A. Anaerobic respiration, B. Substrate level phosphorylation, C. Oxidative phosphorylation via ATPases D, A and B. C. The proton gradient across bacterial membranes is involved in A. Generation of ATP in the presence of an external electron acceptor, B. Active transport of some ions, C. Rotation of bacterial flagella, D. All of the above, E. None of the above. D. Anaerobic respiration encompasses which of the following? A. Assimilatory nitrate reduction, B. Dissimilatory nitrate reduction, C. Denitrification, D. Both, B and C. D. Which fermentation pathway is detected using in the methyl red test when differentiating among enteric bacteria? A. Butyridyl fermentation, B. Mixed acid fermentation, C. Ethanol fermentation, D. Lactic acid fermentation, E. Propionic acid fermentation. B. In the etc. What is being pumped across the membrane? AH plus BECNADHDDADP. A. Which of the following does not occur in the Krebs cycle? A. Redox reactions, B. Production of NADH, C. Production of FAD plus D. Production of carbon dioxide, E. Synthesis, PFATP. C. Which of the following terminal electron acceptors has the greatest potential to provide the largest amount of free energy? A. CO2, B. NO3, CO2, D. Fe3 plus ESO4. C. All of the following are the end products of glycolysis except A. Pyruvate, B. ATP, C. NADD, NADHE energy. C. In the process of blank, ATP is synthesized when excited electrons from chlorophyll P700 pass through a series of electron carriers, then return to chlorophyll P700. A. Photosynthesis, B. Cyclic photophosphorylation, C. Acyclic photophosphorylation, D. Non-cyclic photophosphorylation. B. The series of enzymatic steps that fix carbon dioxide into carbohydrates is called the blank. A. Pentose phosphate pathway, B. Embden-Meyerhof pathway, C. Calvin cycle, D. Embden-Meyerhof pathway. C. What is a feature of a virus with a segmented genome? A. It has multiple chromosomes, B. It is haploid, C. It has a genome composed of separate pieces, D. It must have a DNA genome. C. Which of the following lists the steps of viral activity in the correct order from start to finish? A. Attachment, lysis, penetration, replication, B. Lysis, penetration, replication, attachment, C. Attachment, penetration, replication, lysis, D. Penetration, replication, attachment, lysis. A. D. The genetic code was deciphered in the 1960s in large part by the efforts of A. Avery and Griffith, B. Corona and Nirenberg, C. Watson and Crick, D. Hershey and Chase. C. Which of the following is not a pyrimidine? A. Adenine, B. Cytosine, C. Thymine, D. uracil. A. The genetic code is said to be blank because more than one codon will specify a particular amino acid. A. Ambiguous, B. Degenerate, C. Multiplicative, D. Repetitious. B. T or F. Enveloped viruses may enter their host cells by engulfment within coated vesicles. True. E. What makes up a nucleoside? Nitrogenous base plus pentose sugar. What is the difference between ribose and deoxyribose? Ribose equals hydroxide group on carbon 2, deoxyribose equals hydrogen on carbon 2. What are the nitrogenous bases that make up RNA? A, G, C, U. Which sugar is part of the RNA structure? Pentose sugar. What is the base pairing rules in RNA? AUGC. What are the three types of RNA? rRNA, tRNA, mRNA. All genetic material of an organism. Genome. T or F bacteria cells do not have histones. True. T or F bacteria cells are haploid. True. What is the purpose of fertility factors in bacteria cells? Allow transfer of genes between cells. What is the purpose of resistance factors in bacteria cells? Resistance to antibiotics or heavy metals. What is the purpose of bacteriocin factors? To kill other bacteria. What is the purpose of virulence factors in bacteria cells? Provide functions involved in pathogenesis. Which DNA is usually exists as closed circular, supercoiled molecule associated with basic proteins. Prokaryotic. Why is the supercoiling of prokaryotic DNA important? Allows DNA to fit in the cell, helps control access to DNA to polysaccharides molecules responsible for supercoiling and relaxing DNA X gyrase. Which DNA is linear in shape associated with histones coiled into repeating units called nucleosomes. Eukaryotic. T or F replication of DNA can start anywhere on the chromosome. False. How many hydrogen bonds are between the C and G bases? 3. How many hydrogen bonds are between A and T bases? 2. What is the function of helicase? Unzipping the DNA helix. What is the function of primus? Synthesizing an RNA primer. What is the function of DNA polymerase 3? Adding bases to the new DNA chain, synthesizing complementary strands, proofreading the chain for mistakes. What is the purpose of DNA polymerase 1? Removing primer, closing gaps, repairing mistakes. What is the purpose of ligase? Final binding of nicks in DNA during synthesis and repair. What is the purpose of gyrase? Supercoiling. What is the purpose of SSB's protein? Single-strand bind protein. The manner in which genetic instructions for polypeptide synthesis are stored within genome. The genetic code. Sequence of base pairs in DNA corresponds to the amino acid sequence of polypeptide encoded. Collinearity. What is a codon? Genetic code word specifies an amino acid. Who is credited for the codon? Marshall Narenberg. Up to blank different codons can code for a single amino acid. 6. How many codons specify amino acids? 61. What are the stop codons? UGA, AG, A. What encodes a polypeptide, a tRNA or an rRNA? Gene. Organization of codons such that they can be read to give rise to a gene product. Reading frame. What is the enzyme responsible for the production of the RNA molecule? RNA polymerase. Can both strands of DNA be used as a template in transcription? No. Which strand of DNA is used as a template in transcription? 3-5. Summary of RNA processing. Carries a message for a specific protein. mRNA. Which RNA is 500 to 1000 nucleotides long? mRNA. Which RNA is made up of codons? mRNA. What is used to make polypeptide chain? mRNA. Which RNA is made up of 75 to 80 nucleotides long? tRNA. Which RNA picks up the appropriate amino acid floating in the cytoplasm and transports it to the mRNA? tRNA. Which RNA is the shortest? tRNA. Which RNA strand is the longest? rRNA. Which RNA strand is associated with proteins to form ribosomes? rRNA. Where does transcription start? At the promoter DNA. How many amino acids are there? 20. Where does RNA processing take place? Nucleus. What are the three parts of translation? Initiation, elongation, termination. Which of the following feature of E. coli is important for attachment to host epithelial cells? A. Outer membrane. B. Alpha hemolysin. C. Porin. D. Ki antigen. E. Pili. Pili. O. Antigen found on some bacteria is A. Cell surface polysaccharide. B. Channel controlling substance taken into the organism. C. Flagellar protein. D. Peptidoglycan matrix important for cellular rigidity. E. Part of cell wall lipopolysaccharide. Part of cell wall lipopolysaccharide. Movement of DNA from one bacteria to another through a tubular bridge or pilase. A. Conjunction. B. Transportation. C. Transfection. D. Transduction. Conjunction. D. Treatment of the disease with a broad-spectrum oral antimicrobial dependably eradicates the organism and cures the disease. Characteristics of a bacterial capsule include A. All bacteria have one. B. It is composed of peptidoglycan. C. It plays an important role in protecting a bacterium against phagocytosis. D. It is what causes the Gram-Stain reaction. C. It plays an important role in protecting a bacterium against phagocytosis. C. The ability to have a Gram-Stain result. An important characteristic of mycobacteria is that they are A. Gram-negative. B. Rapid-growing. C. Gram-positive. D. Acid-fast. E. Two of the above. Two of the above. The Gram staining procedure is an example of A. Simple staining. B. Negative staining. C. Differential staining. D. Fluorescence staining. C. Differential staining. T or F. Although developed over 100 years ago, Koch's postulates continue to be used successfully in all known human infectious disease. False. Acid-fast organisms such as Mycobacterium tuberculosis resist decolorization by an acid-alcohol wash because of the high concentration of blank in their cell walls. A. Proteins. B. Carbohydrates. C. Lipids. D. Peptidoglycan. Lipids. T or F. Microbiologists study a variety of organisms, but all are considered members of either bacteria or archaea. False. Which of the following is not a major class of growth factors? A. Amino acids. B. Purines and pyrimidines. C. Vitamins. D. Nucleic acids. D. Nucleic acids. Nutrients can be concentrated from dilute solutions by Polysaccharide. B. Endocytosis. C. Active transport. D. Group translocation. E. Two of the above. Two of the above. Which of the following is a characteristic of active transport? A. Saturable uptake rate. B. Use of ATP or protein motive force as a source of metabolic energy. C. Can move materials against a concentration gradient. D. All of the choices. E. Only B and C are correct. D. All of the choices. T or F. Peptide interbridges are often found in the peptidoglycan cell wall structure of Gram-negative bacteria. False. Which of the following is not part of the peptidoglycan? A. Covalent bonds. B. Core polysaccharide. C. Amino acids. D. Beta 1 to 4 linkage. E. 2 of the above. Core polysaccharide. The lipopolysaccharide that is found in the outer membrane of gram-negative bacteria is also known as A. Exotoxin. B. Endotoxin. C. Murine. D. Brown's lipoprotein. E. 2 of the above. Endotoxin. T or F. Gram-positive bacteria have a structurally and chemically more complex peptidoglycan than gram-negative bacteria. True. T or F. The rotation of bacterial flagella can be powered by only PMF. False. T or F. Gram-negative bacteria are more resistant than gram-positive bacteria to some antibiotics because the gram-negative periplasmic space may contain enzymes that can degrade or modify drugs. True. Studies by Emil von Behring and Shibasa Soto demonstrated that inactivated toxins can induce the synthesis of antitoxins in the blood of rabbits. These antitoxins are the basis of A. Humoral immunity. B. Cell-mediated immunity. C. Antibiotic immunity. D. Phagocyte-mediated immunity. Humoral immunity. Gram stain derives its name from A. Original slots weighing a gram. B. The inventor. C. Unknown source. The inventor. C. What do the outer membrane and the cytoplasmic membrane have in common? A. They are found only in gram-negative bacteria. B. They contain phospholipids. C. They are double-layered structures. D. A and B. E. A, B, and C. B. T or F. Plasmid is only found in bacteria. False. In 1 cm there are A. 10 micrometers. B. 1000 micrometers. C. 10,000 micrometers. C. Who is credited with developing and documenting the first vaccination procedure against smallpox? A. Koch. B. Pasteur. C. Jenner. D. Lister. Jenner. Which of the following is not considered a macronutrient? A. Carbon. B. Calcium. C. Potassium. D. Cobalt. Cobalt. Which of the following bacterial cytoskeleton protein is a tubulin homologue? A. FTSC. B. Rb. C. MID. D. Parm. FTSC. Who of the following was the first to observe and accurately describe microorganisms? A. Pasteur. B. Lister. C. Van Leeuwenhoek. D. Tyndall. C. Van Leeuwenhoek. A. The patient with the pileated strain is ill, the other is not. Basic dyes such as methylene blue bind to cellular molecules that are A. Hydrophobic. B. Negatively charged. C. Positively charged. D. Aromatic. Negatively charged. Which of the following is found in bacterial membrane phospholipids? A. D-glycerol. B. L-glycerol. C. Branched isoprene chain. E. 2 of the above. 2 of the above. T or F. Some archaea are known to cause disease to human. False. T or F. Most endospore-forming bacteria are gram-positive. True. Flexible bacteria with a helical shape are called A. Vibrios. B. Spirilla. C. Spirochetes. D. Cucurbitacilli. C. Spirochetes. Which fermentation pathway is detected using the methyl red test when differentiating among enteric bacteria? A. Butyridyl fermentation. B. Mixed acid fermentation. C. Lactic acid fermentation. B. Mixed acid fermentation. Which of the following does not occur in the Kerb cycle? A. Redox reactions. B. Production of NADH. C. Production of FAD plus D. Production of CO2. C. Production of FAD+. Which of the following terminal electron acceptors has the greatest potential to provide the largest amount of free energy? A. CO2b, NO3c, O2d, SO4. O2. All of the following are the end products of glycolysis except A. Pyruvate B NADHC, NADPD, CO2E, 2 of the above. E. 2 of the above. The proton gradient across bacterial membranes is involved in A. Generation of ATP in the presence of an external electron acceptor B. Active transport of some ion C. Rotation of bacterial flagella D. All of the above E. 2 of the above. D. All of the above. An electron carrier that is used in harvesting energy from glucose molecules in a series of gradual steps in the cytoplasm of bacteria is A. Pyruvate B. FADC, ATPD, NAD plus E. 2 of the above. E. 2 of the above. The most common pathway for conversion of glucose to pyruvate is A. Indnodautoff B. Pentose phosphate C. Embden-Meyerhof D. Mixed acid fermentation. C. Embden-Meyerhof. In the process of blank, ATP is synthesized when excited from chlorophyll P700 pass through a series of electron carriers then return to chlorophyll P700. Cyclic photophosphorylation. T or F. All prokaryotic photosynthesized are anexogenic. False. Which of the following is not true regarding ATP synthesis? A. They require protein motivores to make ATP. B. They span the inner membrane of mitochondria. C. The proton flow is outward during ATP synthesis. C. The proton flow is outward during ATP synthesis. The series of enzymatic steps that fixes carbon dioxide into carbohydrate is called the A. Pentose phosphate pathway. B. Embden-Meyerhof pathway. C. Calvin cycle. D. Indnodautoff pathway. Calvin cycle. In the absence of oxygen, eukaryotic cells are restricted to H. Amosmotic phosphorylation. B. Cyclic photophosphorylation. C. Substrate level phosphorylation. D. Oxidative phosphorylation. E. Non-cyclic photophosphorylation. C. Substrate level phosphorylation. The major function of the TCA cycle is A. Energy production. B. Provision of carbon skeletons for biosynthesis of cell components. C. Direct oxidation of 4 and 5 carbon sugars during anaerobic growth. D. Energy production and provision of carbon skeletons for biosynthesis of cell components. D. Energy production and provision of carbon skeletons for biosynthesis of cell components. During breakdown of fatty acids, carbons are removed blank at a time as blank A. 1. Carbon dioxide B. 1. Methane C. 2. Acetyl-CoA D. 2. Ethanol C. 2. Acetyl-CoA. The decarboxylation of pyruvate produces A. NADHB, acetyl-CoA C, CO2D, ATPE. Only A, B, and C are correct. E. Only A, B, and C are correct. T or F. Bacterial electron transport chains may be comprised of fewer components and have lower PO ratios than mitochondrial transport chains. False. Which of the following is normally treated by topical application of antifungal drugs? A. Systemic mycosis. B. Superficial mycosis. C. Subcutaneous mycosis. B. Superficial mycosis. The product of fermentation which is most important for the cell survival is A. NADHB, ATPC, alcohol D, CO2E, NAD+. NAD+. B. It may have the normal bases found in prokaryotic and eukaryotic DNA, or it may have one or more unusual bases. The standard reduction potential of a redox reaction is a measure of the tendency of the blank-to-blank electrons. A. Reductant, gain. B. Reductant, lose. C. Oxidant, gain. D. Oxidant, lose. B. Reductant, lose. Which of the following is true regarding pentose phosphate pathway? A. NADPH is produced in this pathway. B. Source of 4 and 5 carbon sugars for many purposes. C. Intermediates may be used to produce ATP. D. All of the above. E. 2 of the above. D. All of the above. A blank assay is most useful for determining the viability of a viral preparation. A. Direct count. B. Hemagglutinin. C. Plaque. D. PCR. C. Plaque. All of the reactions of glucose oxidation that follow glycolysis involving the transfer of electrons to their final acceptor, oxygen, take place in eukaryotic cells in the blank mitochondria. Fats undergo a process called blank oxidation in which the products are acetyl coenzyme molecules. Beta. Non-capsid proteins that aid in the assembly of virion structures but do not become incorporated into the virion are referred to as blank proteins. Scaffolding. A bacterium carrying a dormant phage inserted in the chromosome is called a blank. Lysogen. Viruses that are polyhedrons with 20 sides are said to have blank symmetry. Icosahedral. Organisms that directly reduce almost all of their pyruvate to lactate are called blank fermenters. Homolectic. Blank are glycoprotein spikes protruding from the outer surface of the viral envelope. Peplimers. During replication, the two strands of the DNA molecule are unwound from one another by enzymes called A. ligases, B. helicases, C. primases. Helices. The Y-shaped part of the DNA molecule where the actual replication process occurs is called the replication blank. A. Tongue. B. Fork. C. Splice. Fork. Each DNA molecule has one or more blank site where replication begins. A. Promoter. B. Replicon. C. Origin. Origin. In the eukaryotic chromosome, the DNA is organized into blank in which the DNA is twisted around small basic proteins that are rich in the amino acids lysine and arginine. A. Nucleosomes. B. Replisomes. C. Dictyosomes. Nucleosomes. When a DNA molecule is replicated, the daughter molecules contain one strand of parental DNA and one strand of newly synthesized DNA. This is called blank replication. A. Semiconservative. B. Conservative. C. Semiconservative. Semiconservative. Which of the following is not a pyrimidine? A. Adenine. B. Cytosine. C. Thymine. D. Uracil. Adenine. The coding sequence in the DNA of blank is normally continuous, that is, it is not interrupted by non-coding sequences. A. Prokaryotes. B. Eukaryotes. C. Both prokaryotes and eukaryotes. D. Neither prokaryotes nor eukaryotes. Prokaryotes. Spontaneous mutations are caused by A. Errors in DNA replication. B. Insertion of transposes. C. Either errors in DNA replication or insertion of transposes. D. Neither errors in DNA replications nor in C. Either errors in DNA replication or insertion of transposes. Microbial strains that can grow on minimal medium are called blank. A. Autotrophs. B. Auxotrophs. C. Prototrophs. D. Minitrophs. C. Prototrophs. Which of the following is not a termination stop codon? A. Aug. B. Ag. C. Ag. D. Yuga. A. Aug. Mutant strains that are unable to grow on minimal medium but require that the medium contains an adequate supply of the end products of a metabolic pathway are called blank. A. Autotrophs. B. Auxotrophs. C. Prototrophs. D. Mentrophs. B. Auxotrophs. The pentose phosphate pathway provided A. ATP. B. NADPH. 4 and 5 carbon sugars for amino acid and nucleic acid synthesis. D. All of these. All. The latent form of a bacteriophage genome that exists when a phage establishes lysogeny is called blank. A. Lysogen. B. Prophage. C. Viro. D. Prion. B. Prophage. Why is it so hard to treat viral infections? Viruses use the metabolic machinery of their hosts, which limits selective points of attack. Antibiotics that are given by injection are said to have blank mode of administrations. A. Oral. B. Venal. C. Parenteral. D. Topical. C. Parenteral. Moist heat sterilizes by A, causing the formation of thymine dimers. B. Denaturing protein. C. Causing production of singlet oxygen. D. All of these. B. Denaturing proteins. Organisms that grow well at 0°C and have the optimum growth temperature between 20°C and 50°C are called A. Psychotrophs. B. Psychrophiles. C. Mesophiles. A. Psychotrophs. T or F. Cell walls of archaea and bacteria contain peptidoglycan. False. T or F. Archaea membranes have a typical bilayer structure. False. Tachoic acids are found in A. the cell membranes of archaea bacteria. B. the cell walls of gram-positive bacteria. C. the cell walls of gram-negative bacteria. D. the cell membranes of all prokaryotes. Two of the above are correct. B. the cell walls of gram-positive bacteria. Which of the following is true of all viruses? A. They contain DNA. B. They contain RNA. C. They result in the direction destruction of their host. D. They contain nucleic acid. E. None of the above is true. D. They contain nucleic acid. Utilizing carbon dioxide or nitrate as terminal electron acceptor. Which of the following are involved in alcoholic fermentation either as products or reactants? A. Ethanol. B. Lactic acid. C. Carbon dioxide. D. Both A and B. E. Both A and C. E. Both A and C. C. They grow best at temperatures above 45°C. The product of fermentation which is most important for the cell survival is A. NADH. B. ATP. C. Alcohol. D. CO2. E. NAD+. ATP. An increase in magnification causes A. Decrease in the field of view. B. Decrease in the working distance. C. All of the above. D. None of the above. All. T or F. In the process of fermentation, an organic substrate such as glucose is reduced and an intermediate of the pathway such as pyruvate is oxidized. False. How are archaeal, bacterial, and eukaryotic cells alike? A. They lack nuclear membrane. B. Mitochondria are present. C. Chloroplasts are present. D. They have cell membrane. E. They all contain histone proteins. D. They have cell membrane. T or F. The rotation of bacterial flagella is powered by ATP hydrolysis. False. If you had a defined medium, which choice, when added, would make the medium complex? A. 5 g of NaCl. B. 10 ml of 100% ethanol. C. 0.1% of pure lactose. D. 5 g of beef extracts powder. E. None of the above. D. 5 g of beef extracts powder. T or F. A drug that disrupts a microbial function not found in animal cells usually has a lower therapeutic index. True. Which of the following statements concerning peptidoglycan is true? A. It occurs only in bacteria. B. It is sometimes found in archaeal cells. C. It contains a peptide portion and glycan portion. D. All of the above are true. E. Only A and C are true. E. Only A and C are true. Pseudopeptidoglycan occurs in A. Bacteria. B. Mycoplasma. C. Archaea. D. Mycobacterium. E. Two of the above. C. Archaea. T or F. DNA gyrase is a type of topoisomerase found in prokaryotic cells. True. T or F. The Embden-Meyer's pathway represents the major means of glucose catabolism in most cells. True. T or F. Substrate-level phosphorylation couples a chemical reaction with the generation of ATP. Oxidative phosphorylation uses the proton motive force to generate ATP. True. Which events are parts of the tricarboxylic acid cycle? A. Citrate reacts with oxalostate to form acetyl-CoA. B. Glucose is converted to fructose. C. CO2, H20, reduced coenzymes, and ATP produced. D. Free O2 is liberated. E. NAD is regenerated. C. CO2, H20, reduced coenzymes and ATP produced. Which of the following best describe possible energy sources for a chemoorganotroph? A. Fructose. B. Glucose. C. H2S. D. All of the above. E. 2 of the above. E. 2 of the above. C. They are required for host growth and/or reproduction. Which of the following is associated with endotoxin? A. E. coli. B. Symptoms of fever and shock. C. Salmonella species. D. LPS molecules. E. All of the above. D. LPS molecules. Which of the following was lysed by penicillin? A. Mycoplasma sp. B. Eukaryotic cells. C. Non-resistant Staphylococcus sp. D. A and B. Non-resistant Staphylococcus sp. Which are features of the outer membrane? A. It is a lipid bilayer. B. It contains phospholipid. C. It prevents drugs from reaching the cytoplasm. D. A and B. E. A, B, and C are correct. A, B, and C are correct. T or F. The non-coding sequences located between the start codon and the stop codon in interrupted genes are called introns. True. Post-transcriptional modifications that yield eukaryotic mRNAs include which of the following? A. Splicing to remove introns. B. 5' polytracks. C. 3' caps. D. All of the choices. Splicing to remove introns. The strand of DNA for a particular gene that is copied by the RNA polymerase to form mRNA is called the blank strand. A. Leading. B. Template. C. Transcription. D. Copy. Template. The enzyme sequentially that links adjacent amino acids into the growing polypeptide chain. A. Ligase. B. Peptidyltransferase. C. Lyase. D. Phosphorylase. Peptidyltransferase. Which of the following is not considered the final product of the expression of a gene? A. A polypeptide chain. B. An mRNA molecule. C. A tRNA molecule. D. An rRNA molecule. An mRNA molecule. When a recipient cell requires a piece of naked DNA from the environment, it is called A. Conjugation. B. Transformation. C. Transduction. D. Translocation. Transformation. Transfer of genetic information via direct cell-cell contact is called A. Transformation. B. Transduction. C. Transfection. D. Conjugation. Conjunction. T or F. Point mutation is not always deleterious. True. Which of the following organisms has no motile cells at any point of their life cycle? A. Protozoa. B. Algae. C. Fungi. D. 2 of the above. Fungi. Which is not true of viruses? A. They are obligate intracellular parasites. B. They can be destroyed with antibiotics. C. They can be observed with an electron microscope. D. They are cellular. E. They replicate only inside host. They can be destroyed with antibiotics. T or F. Algae are simple eukaryotic photosynthetic organisms using chlorophyll as primary pigment in oxygenic photosynthesis. True. D. Although penicillin inhibits peptidoglycan synthesis, bacterial cells will continue to grow normally in the presence of penicillin in an environment. A. Hypotonic. B. Isotonic. C. Hypertonic. D. Nonpolar. Isotonic. Microorganisms are most nearly uniform in terms of chemical physiological properties during blank phase. A. Lag. B. Exponential. C. Stationary. D. Decline. Exponential. Which of the following processes can be used to bring nutrients in a cell against a concentration gradient? A. Active transport. B. Facilitated transport. C. Active transport and facilitated transport. Active transport. T or F. Copper is considered a micronutrient. True. T or F. The size and shape of a colony is not useful for identification of a microorganism because most microorganisms produce colonies that are indistinguishable from one another. False. T or F. Although similar in function, the eukaryotic ribosome is generally smaller and more complex than the prokaryotic ribosome. False. Moist heat sterilization at 100 degrees Celsius kills all the following except A. Bacterial endospores, B. Fungal spores, C. Viruses, D. Bacterial endospores and fungal spores. Bacterial endospores. T or F. The effects of most antimicrobial drugs are greater if the microorganisms are actively growing. False. T or F. For most genes, coding information is found in both strands. True.